Michelle, or Hannah, I will defer to you on when we should go ahead and get started.
I just sent the link to Zev to hopefully get him in quickly, but we are pretty packed. So if you feel comfortable jumping in, I think that'd be great to get started.
Yes, happy to do that. Um, and I will, when maybe at the end or whenever Zev comes on, he can also give a brief introduction. But I'm happy to start our first day here of this workshop focused on synthetic and systems approaches to interrogate spatiotemporal processes in cancer. This is a workshop, the first of 3 workshops, organized really very beautifully by several NCI staff who are working diligently and furiously in the background to keep everything running smoothly today and all of us on time. But it's really been a really interesting experience to put this together with them and really think about some of the key developments that have occurred in the past few years in both of these fields that really we think put us in a great position to think about how these 2 fields may be brought together to really advance our understanding of cancer. We do have 3-day workshop. First day will focus on transformation. And just to jump in on here. Oops, excuse me. So first, we do have a very mixed audience today. So some of you identify as systems biologists, some as synthetic biologists, and maybe others as neither. And so what we wanted to do first was to just give a little bit of a very high level overview of these 2 different fields. And again, these are disclaimer, these are views of the co-moderators of Zev Gartner and myself for how we would define systems and synthetic biology, where systems biology is really designed to integrate experimental and computational approaches to study cancer as complex systems. And synthetic biology is using engineering principles to design biological systems. And we also really think about systems biology as an approach that lets us generate new testable hypotheses that can shed light on mechanisms underlying disease progression, therapeutic response, and so on. And in contrast, synthetic biology really has developed a very large tool set that lets us test these hypotheses in various ways. So with that as a high-level background, why is this now the right time for a workshop on these 2 topics? And we posit that is for 2 primary reasons. First, that there are recent advances in both spatial and temporal measurement technologies. For instance, things like multiplex tissue imaging, spatial transcriptomics, intravital imaging, approaches of lineage tracing and cell recording that let us look at dynamics of cells, both in very short time frames as well as much longer time frames when we're thinking about lineage tracing. Optogenetics and cell engineering that let us manipulate biological systems in different ways, and so on. Over the course of the day, we'll be hearing about a number of different techniques that really allow us to dig deep into underlying biological mechanisms. And so what we have in the next few days is really thinking about how we can bring some of these tools and approaches together to address questions of transformation, which will be today, tumor microenvironment on Wednesday, and then therapeutic resistance in the 3rd workshop next week. And one of our major goals in these 3 workshops is to foster idea exchange across disciplines. And so we really want to frame some of our discussions, in particular when we move into the breakout areas, in thinking about what are the open questions in cancer transformation, what don't we know that we think we may be poised now to begin to gain insights into, what opportunities exist with some of the expertise and approaches that are now available, and then finally, what challenges remain. There's a room full of scientists. We know although there are opportunities, there are always always challenges that come along with them that we need to be thinking about in terms of future questions or future steps. But really what we are hoping in these meetings is to really allow people to think deeply about these things, to speak up and share their ideas with one another. And toward that goal, the NCI staff has also put together a number of tools that will allow us to interact with each other in real time and actually share notes in electronic format. So the Miro board is the tool of the day. And there is now in the chat a link to that Miro board. In a few moments, I'll stop. And so the NCI staff can tell me if it needs to be reposted or not. But within there, you can actually log. Okay, thank you, Michelle. She just posted the link again. Feel free to go in right now, take a sticky note, introduce yourself, write your name, perhaps where you're from, and also some note about your primary discipline or interest. And then as we move through the day, we will have a few opportunities to have some focused discussions around key topics. And so we have now identified a few different Miro board locations to help capture notes and thinking about those different conversations. And with that, I will close there and encourage you all to really participate wholeheartedly with us today. And with that, I also think if Zev and perhaps Shannon Hughes are on. And in fact, as well, I'm going to take the liberty as the one who has the stage to also ask if possible, this is a change of schedule, for Hannah and Michelle to unmute your cameras, just so everyone can see who's really the team behind putting this together. I feel like it's, in a perfect world, we would all be in the same room together, but we're in our virtual spaces today. So thank you both for organizing this and taking the lead to bring together these groups. Two great program staff. And as well, I don't see everybody on my. Is Shannon going to say a few words as well?
Excellent. I was going to say a few words, Laura.
Excellent. All right, I will hand over to you now.
Thank you. And I want to start by first thanking Laura and Zev for working with our just excellent program staff, Hannah, Michelle, and others. There's a big team behind this particular event. I think you'll find the agenda to be exciting and also different from other meetings that you've gone to recently. And I encourage people to use the Miro board. And I can see it out of the corner of my eye, lots of names flying around on the Miro board. So I won't take much of your time. I'm Shannon Hughes. I'm the Deputy Director of the Division of Cancer Biology here at NCI. And on behalf of everyone at the NCI, I want to welcome you to this virtual workshop series. I'm not sure how many of you were able to attend either in person or virtually the American Association for Cancer Research meeting this year, AACR. But one thing that you saw either online or in person was that spatial biology is everywhere. It has really hit the mainstream in cancer biology and is headed towards the clinic. This is true through both of the number of plenary speakers that we saw deriving insights from spatial relationships within tumors, and also from the number of vendors within the exhibition hall who were selling new technologies and ways for us to continually measure things in a spatially conserved context. But in reality, we really are a little late to this game. Molecular biologists have been thinking about the spatial organization of chromatin and how that results in gene expression for a long time. Developmental biologists have been thinking about the patterning of cells and how gradients induce different behaviors within tissues. And ecologists have been thinking about the population dynamics and how populations move in space and in time. So we're here today to talk about how we can take advantage of these types of insights that we're gleaning from a number of NCI and other supported programs, such as the Cancer Systems Biology Consortium, the Physical Science Oncology Network, Human Tumor Atlas Network, the great program that has supported developments in synthetic biology lately, and how we can really interrogate and test some of these spatial relationships that we're finding. We now have a very large number of hypotheses that could be tested. And importantly, many of these hypotheses are derived directly from human tissue, where we know we can't interrogate them in real time, at least not in much of an ethical manner. So how do we think about validating these types of hypotheses or testing them using new systems, new computational approaches, new experimental approaches, taking advantage of automation, controllable systems such as those that are elegantly being built by synthetic biologists, and importantly, doing that within a cancer context over time so that we can measure the dynamics. So I think this is a space where we want to invite you to be brave and to embrace the complexity and maybe leave some of your previous thoughts of what's possible in these spaces at the door as you enter this virtual environment and interact on the Miro board. Because this is really a place that is poised to make an impact in patients in the near future. So welcome, we're really excited for everyone to be participating in these virtual workshops. We're especially thankful for the folks who are providing keynote talks. Today we'll hear from Ken Lau. We'll also hear from Thea Tilsty and Gordon Mills in the future. All three of them are willing to make controversial and debatable statements around biology that I think will be picked up in the further conversations and the design of this workshop. So have fun. Thanks again. And I see Zev, so I'm just going to pass it back to Zev. He's the only person I can see on the camera. So.
Thanks, Shannon, and thanks everyone for organizing this exciting series of workshops. I don't have much to add. Just want to introduce myself and say hi, and let's get the ball rolling.
So I'm not sure, Michelle or Hannah, if one of you is going to take it from here.
I think we are ready for Ken. Excellent, to start sharing. I will do just a brief introduction of Dr. Ken Lau, who is a professor at Vanderbilt University in the Department of Cell and Developmental Biology. He is also leading efforts in computational biology at Vanderbilt and has been involved in the Cancer Systems Biology Consortium for a number of years. He's using a variety of really cutting-edge, state-of-the-art experimental techniques to examine cancers as complex systems. And with that, Ken, I will hand over to you.
Thank you. So can you hear me and can you see my slides?
We can hear you. We saw your slides, although I don't see them anymore, and I don't.
Okay.
Your camera is also not on.
Okay, hold on a second. Okay, my camera is on, right?
Excellent. Yes. Yes.
And my slides. Can you see my slides?
Yes. Not yet in presenter view, but we do see them.
Okay.
Perfect.
Are we good?
Yes.
Okay, great.
Great. Thank you so much.
Well, thank you, Laura and Seth, for inviting me to give some controversial points about tumor initiation. So, my lab works at the interface between damage and inflammation and pre-cancer. And as Shannon said, there are new technologies now where we can, profile the spatial aspect of tumor biology, specifically the tumor microenvironment. A lot of these technologies, though, would give you sort of snapshots in time. And even if you longitudinally sample an individual patient, the events that you'll see are mostly independent events, right? So, our lab is, very interested in looking at the temporal aspect, which allow us to look at the origins, the order of events, as well as the persistence of these events. And it's sort of, brings me back to my systems biology training, right? Where, building dynamic models were very important. So I'm going to do my talk a little bit differently from before. I'm just going to thank the people first, because I think I'm going to end with some rather controversial, maybe some negative data, and then we can open it up for discussion. So, I run a small lab of less than 10 people, but I think this is sort of the overall theme of doing systems biology as well as synthetic biology, is that it's a team sport. So, okay, so can people see the slide? Because someone said they cannot see the slide. So,
I can see the slide.
okay, okay.
Thank
So
you.
as I was saying, it's sort of a team sport. So we're a small lab, but embedded in this large village within Vanderbilt. We're getting human specimens as well as experimental validations, bioinformatics, etc. So today I'm going to talk about, some of the vignettes of work that are done by various graduate students in my lab. So I think Laura gave a very good introduction of systems biology versus synthetic biology. And I'm just going to reiterate, systems biology is kind of like a philosophical view, right, of looking at biology as a whole. But when you get down to it, the approach is really you're taking large data sets or large number of observations and putting those data sets or those observations into sort of a quantitative framework, quantitative mathematical framework. So this is kind of akin to engineering, building a model, right, of how a car works, for example. So the models that we use are, mechanistic modeling, data-driven modeling. Nowadays, there's AI. So that would give you, for the most part, at the end of the day, predictions and correlations, whether whether you model the data with a simple linear regression model or a very complex dynamic partial differentiation equation model, right? But what we need in addition to correlations, and as Laura and Shannon talked about, is we need systems biology would allow us to generate hypotheses that are perhaps hidden and emergent in a complex network, right? But we still need validation in terms of experimental models where we can break down the system to understand how different parts work, right? So the models that we use, right, in terms of testing hypotheses from systems biology studies are, cell lines, in vivo models like mouse models and organotypic models, right? So, so this cycle, right, of systems biology, modeling, and then experimental validation. So we collect data, we build a model, then we simulate the model so we can make predictions, and then we validate these. We go back into the experimental system to test sort of these hypotheses. But for today's workshop, we're interested in spatially defined cancer processes, right? So the experimental models that we are interested in looking at must be able to address these questions in spatial biology, which means we need models that are not a lawn of cells, of cell lines, but comprises of different cell types organized in space. So this is, a primer for the synthetic biologists in the audience. And the current models that we have are the in vivo models, like the mouse models. So this is what my lab is based on, focus on mouse models. But we have been trying to get away from mouse models for the past 3 years. And then you'll see where we are at the end of the presentation. Then there are these 3D organoid models embedded in Matrigel where you can co-culture tumor cells with the microenvironmental cells like immune cells. And then more sophisticated models focus on 2D monolayers, culturing organoids in monolayers, including air-liquid interface, where we can co-culture the tumor epithelial cells as well as the different components of the microenvironment in different spatial compartments, the apical side versus the basal lateral side. Then there are explants and slice cultures, organs on a chip where you control the flow as well as the presence of different compartments in the different channels of the system, as well as pattern matrices where we can bioengineer different configurations of the ECM to look at this, how different structure of the tissue affects the biology of the cells. So today I want to sort of discuss concepts of data and models as applied to tumor initiation, specifically giving you examples of us trying to go back and forth between systems modeling and sort of experimental functional validation using some of these models. So tumor initiation. So why do we want to study tumor initiation and pre-cancer? Actually, we have been doing this for a long time, but now it is, again, an emergent topic in cancer biology. So just to give you an example, early onset cancer, the age of onset of cancer is a significant topic of discussion nowadays. So there has been an increase in incidences of early onset, for example, colorectal cancers. And the time when a tumor starts, right, is essentially a tumor initiation problem as well as a temporal problem. So now there has been more interest in looking at the pre-cancer state. So there's hallmarks of pre-cancer perspective recently published as a companion piece to the hallmarks of cancer. So how do we define a pre-cancer, right? If you ask a pathologist, a pre-cancer is something that is morphologically different, histologically different. You can look at it and you know that it's different from normal tissues. However, there are other descriptions or other definitions of pre-cancer, right? So for example, this model proposed by Bill Grady and colleagues, what are they looking at is they're using epigenetic drift to track the timing of the original event where the first abnormal cell arose. And what they found was that the first event occurred between 10 to 20 years of age in humans. So occurs very early. So at that point, the abnormal cells actually look normal. They're morphologically normal, but molecularly abnormal. And this is work by Peter Campbell, Mike Stratton, and others in the esophagus. And again, looking at morphologically, histologically normal esophagus, but you can actually detect mutations, specifically mutations in driver genes that clonally expand over time, right? Again, documenting that when we talk about tumor initiation, we have to think back to the earlier stages where the cells are molecularly abnormal, but morphologically normal. So with that said, we need to know what we mean by molecularly abnormal, right? So what would that cell look like molecularly? So we started this study a while back at the start of the Human Tumor Atlas Network, and we were quite surprised at that time. There were no large-scale TCGA profiling efforts done on precancers, right? So that's sort of the study that we engaged in. We did single-cell sequencing on, at that time, maybe one of the largest cohort of samples. So we did 60 precancerous polyps in the colon, coupled to the adjacent normal tissues, or 120 data sets. So this was back in the day before 10x was a thing. So with single-cell genomics, let me get my pointer here, is that we were able to identify epithelial cell populations that are specific to the specific precancer subtypes, right? The adenoma-specific cells, as well as the serratus-specific cells, specific to the serrated polyps, so the two subsets of colorectal precancers. So just because this is published a while back, I'm just going to summarize. Well, the conventional adenomas, which are thought to be the precursors, not 100%, mostly the precursors for the microsatellite stable colorectal cancers, they are basically super stem cells. driven by Wnt signaling, and they are thought to originate from the bottom of the crypt, the normal crypt-based columnar stem cells. And with regards to the serrated polyp-specific cells, they are more interesting. So they are thought to be the precursors of these hypermutated microsatellite unstable colorectal cancers. And instead of having a lot of stem cell capacity, instead they arose from what we call pyloric metaplasia program, stemming from loss of colonic identity and fetal reversion. So these cells, they no longer look like colon, but now they look like the stomach. They look like a gastric epithelium. And their cell of origin is thought to be the surface, the non-crypt-based stem cells of the colon. So this one is driven by stem cell expansion, and then the other is driven by cellular plasticity. So given that now we have a molecular profile of how these abnormal cells look like, we can then, one more thing is, the location of these polyps are also different, right? Conventional adenoma, they're mostly detected in distal part of the colon, serrated in the proximal part of the colon. So given that we have a molecular picture of what these abnormal cells look like, we can then go back to look at normal tissues to look at whether these cells are present, right? So using MUC5AC as a marker, we can look at a lot of normal cases, but we do find these abnormal cells sporadically, rarely, within these morphologically normal colons, and again, at the top of the crypt surface and not down in the crypt. And these cells increase in the context of IBD, in the context of damage, right? And this is another single cell RNA-seq data set we put out recently. 60 patients, TI, terminal ileum coupled to ascending colon. And just to keep a long story short, if you only look at the ascending colon, you can find inflammation-specific epithelial cells. And again, these cells are characterized by loss of colonic identity, loss of CDX2, and upregulation of the fetal reversion type of gene signature, but not yet gastric metaplasia. So they lost their identity, they adopt the fetal identity, but then they've not yet transitioned to a gastric identity. So these can be thought to be sort of these pre-cancer cells that are molecularly distinct. So we can sort of kind of understand now there's an intimate relationship between damage and repair, as well as, at least in the serrated case, tumorigenesis. And of course, when we talk about damage and repair mechanisms, it's always accompanied by a change in the tumor microenvironment, right? Either through fibrosis or inflammatory response, right? So if we look at the tumor microenvironment between the serrated adenomas, serrated lesions, as well as adenomas, we can see a lot of infiltration of these CD8 positive cytotoxic T cells, which are like high value targets for immunotherapy. But in the field, right, we thought that infiltration of CD8 positive T cells would be a function of hypermutation and neoantigen expansion. So we expected that these serrated lesions, because they are the precursors of these hypermutated MSI high colorectal cancers, to have a hypermutation phenotype. But what we saw was that none of them presented a hypermutation phenotype. So that led us to think about other mechanisms by which these abnormal epithelial cells can communicate with the immune system to drive this cytotoxic immunity. So this is how, when we sort of switch back to go into the mouse model to address this very specific question. So we thought there's differences in stemness between the two types of polyps, that if we can modulate stemness in the tumor cells, we can impact T cell interactions and antigen presentation. So what we did, we took a mouse model and we drove tumorigenesis using the same mutation, APC mutation, but from different cell populations, from stem cells using LRIC1, we call these LRIC1 tumors, or from non-stem cells, we call those MIS1 tumors. And luckily, we were indeed able to modulate the degree of stemness based on these organoid assays, right? So the LRIC1 tumor cells can readily form organoids, whereas the MIS1 tumor cells cannot. And then we also saw that the MIS1 tumors has a much richer infiltrated CD8 positive T cell infiltration into the tumor microenvironment. And then when we take these cells ex vivo in a organoid T cell co-culture assay, that is dependent on over-antigen presentation. We saw that the MIS1 tumors, the tumors that have less stemness driven by non-stem cells, can drive naive T cell proliferation in an antigen-dependent manner, as opposed to the stem cell-driven LRIC1 tumors. A second model that we use, that model, we drove the tumors with the same mutation from different drivers, different cells. Here, we drove mutations from the same cells, but using different mutations. APC, which are the characteristic mutation for the conventional adenomas, and BRAF. So what we see, right, with APC loss, we get basically a big ball of stem cells, right, driven by Wnt signaling. But this is characterized by absence of CD8 T cell infiltration. Whereas for the BRAF tumors, we don't get a tumor per se, but we start to get this change in cellular identity. We actually see villus form in the proximal colon of the mouse. Right? Usually, these villi are characteristic of the small intestine and not of the colon. And more importantly, we saw that there's increased infiltration of these CD8 cells into these differentiated villi, right? And what's more important is that the MHC class 1 as well as class 2 is upregulated only in these differentiated villiform metaplasias in these mice, as opposed to the APC null mice where the MHC class 1 and class 2 antigen presentation machinery is downregulated. So, this speaks to, hey, looking at the properties and the identity of precancer cells as related to how they modulate the tumor microenvironment. So next, we want to look at what about the transition from the precancer to cancer. So this is work driven by an undergraduate student in the lab, and the approach here is to take what we call phylogeography approach, is to select cancer samples that are spatially heterogeneous with multiple morphology characteristic of normal, precancer, and cancer within the same lesion, the same cancer. And what we did was we used some old school spatial genomics approach, laser capture microdissection, to look at mutations as well as copy number changes. So here we use shared and unshared copy number changes as mutations to reconstruct a tumor evolutionary phylogeny on a per tumor basis, okay? So you can sort of imagine that this has some temporal component to it, right? Given that where these regions branch off in this phylogenetic tree would denote, the timing by which these molecular events occur. So we can actually order the different regions by using this sort of temporal scaffold defined by genetics. And because we couple this sort of temporal genetic approach with spatial transcriptomics, we can also then understand what are changing with regards to gene programs of the tumor cells, as well as what's happening in the tumor microenvironment. So again, this is published, just to keep a long story short. What we did was we did a progression pseudo-time analysis. So it's kind of like a pseudo-time analysis, but the progression is scaffolded, is anchored by the genetics of progression. And then we can overlay sort of gene programs and microenvironmental programs that sort of correlate to tumor transition from precancer to cancer. And what we saw was sort of typical, right? We saw, as a function of progression, we saw increases in ICMS2 stemness and hypoxia. But also what's interesting is we're focused on this, what we call the immune exclusion program, that are comprised of 4 genes that are epithelially tumor cell expressed, but modulate the extracellular matrix. So these genes get upregulated during that precise transition between precancer and cancer. So the model is kind of looking like this, where during this transition, you get a much denser fibrotic extracellular matrix environment, which excludes cytotoxic T cells, but also allows immunosuppressive neutrophils to get into the tumor. So again, we switch back to these model systems to sort of assess the function of each of these immune exclusion proteins, which are tumor expressed, and their effect on the tumor microenvironment. So what we first focused on, which to go over briefly, is DDR1, which is discoid domain of receptor 1, which is a tyrosine kinase, but it is secreted by tumor cells as an extracellular domain, which is cleaved, the extracellular domain secreted in these novel nanoparticles called the supermirrors. So we collaborate with Incendia. We have a clinical trial actually happening at Vanderbilt University Medical Center. But here, what we did was we took patient specimens, which are explants, we grew them in an air-liquid interface, which preserves the tumor microenvironment. Okay, and then what we did was we administer the therapeutic antibody against DDR1, which neutralizes DDR1 into these tumor explants. And what we saw was that we now start to get now infiltration of these CD8 positive T cells back into the tumor proper. And we coupled this with a larger TMA cohort, as well as a DDR1 knockout colorectal cancer model. Okay? And then the second molecule that we focus on is DPEP1, which is secreted by the tumor cells in the exosome fraction, and now different nanoparticle fraction, which is, so DPEP1 itself in this Cell paper was shown to be able, DPEP1 does many things as a dipeptidase, but one function is that it acts as a physical binder of neutrophils. And from a phage display screen, these individuals here identified this alsol peptide, so this sequence here, which competitively binds to DPEP1 and kicks off the neutrophils. So what we do here is that we took cell lines as a model. So we have SW480 and SW620, which are basically from the same patient that models early and advanced colorectal cancer. And then you can see that in the neutrophil adhesion assay that the SW620 cell, more advanced cancers, bind more neutrophils. And then you can appreciate this difference in scale here, that upon alsol peptide binding, that the SW620 cell line basically have a decreased adhesion of neutrophils. And more importantly, when we go back to a AOM-DSS model of colorectal cancer, if we knock out DPEP1 in this model, you can see that now there's an increase in infiltration of cytotoxic T cells into the tumor proper. So that we now also have studies ongoing administering immunotherapy to this model, and it seems to be quite promising. So the model here is that, in an immune excluded environment, there would be also, as opposed to excluded T cells, there are also immunosuppressive neutrophils that would displace the cytotoxic T cells from the microenvironment. And this is actually modulated by the tumor cells. And this interaction can be perturbed by alsol peptide as well as other pharmacological agents that we're working on right now, which allows the CD8 T cells to infiltrate back into the tumor proper. So these are like multiple vignettes of how we, go from sort of spatial single cell sort of transcriptomics data back to, these models that we can perturb to address specific biological questions. So we talked about tumor initiation. So what do we know about tumor initiation prior to looking at pre-cancer, looking at cancer? So, there's this Fogelstein model, right? That is very stepwise. So you have a single cells accumulating driver mutations, and then as they accumulate more and more driver mutations, then you transition from normal to precancer and precancer to cancer, right? And then, we talked also about driver genes and how they are identified. A lot of the times it's through a statistic approach looking at the frequency of mutations. So you have more APC mutations in a large cohort of cancers compared to KRAS, so it must come earlier. And then you can do this sort of in a more sophisticated fashion, so for example with PCOG, to reconstruct this tumor evolutionary phylogenies using large-scale genomic data. And the implication here is that a single, you'll get these truncal mutations, right? So a single normal cell is the cell of origin of cancer, specifically a normal stem cell, right? And in their paper, they talked about they discarded all the polyclonal trees because irrelevant, right? So we know, for example, that somatic mutations, believe it or not, do occur in normal tissue, so over age, right? So in the colonic crypts, again, this is Trevor Graham, Peter Campbell, Mike Stratton. They look at normal tissues, and we saw that somatic mutations are accumulated over ages, and then we can leverage mutation information, right, to track precancer to cancer, right? And then we thought that because we have mutations in normal, we can also leverage these mutations from normals to determine the relationship between normal tissues and precancers. So that's the question. Problem is, this is extremely difficult to do because in normal tissues, somatic mutation rate is extremely low. So very few number, very sparse data, right? So it's very hard to do, these studies here. But what we can do, as Laura alluded to previously, is that we can use CRISPR-based approach in mouse models to generate synthetic mutations in an accelerated manner, right? So we can do this in specific barcodes of genomes of mice, right? And then, so these barcodes sort of exist outside of the coding genome. So they are sort of in non-coding regions. So they're not selected for, so they only act as recorders. So we have these accelerated synthetic mutations occurring in these barcodes. Then we can perform the same type of phylogenetic and clonality analysis as in normal cells. So this is, again, driven by another student, Marisol Islam, in the lab. So there are other ways where we can do this. Most of the ways in the field that do this in vivo is they would engineer a barcode that can be captured by these poly-A single-cell approaches. So what we did was to go reverse. What we did was we designed a new single-cell platform that can capture the guide RNA and then read out the sequence of the native guide RNA at single-cell resolution, as well as capturing the transcriptome at the same time, right? So within the droplet, you can capture both the guide RNA and the transcriptome. So we call this NSC-seq because we're capturing the native SC guide RNA. So the application of this is actually through these Perturb-seq-based CRISPR screen. Again, most of the Perturb-seq development has been focused on engineering these guide RNA libraries that can be captured by poly-A-based single-cell platforms. But because we can capture the native guide RNA, we can use these common guide RNA libraries like Brunello and Brie. So here is just a proof of concept. You have a parental cell line EPH4, looks like a blob when you do single-cell RNA-seq because relatively homogeneous. But when these cells are infected with a guide RNA library, then you can see structure. And then, the function of these cells can be defined transcriptomically, right? So the cluster of these cells would define the identity of the guide RNA that would hit the same pathway, like the Rho pathway, for example. So this works really well. So for canonical CRISPR, we're expressing the guide RNA from one locus, and then the guide RNA would hit another locus, right? But we can use a homing CRISPR approach where the guide RNA is expressed from the one locus and then hits itself. So you can see that the guide RNA can edit a sequence, what we call a barcode, upstream of the pan, and over multiple cycles, then we have self-mutation as well as a cumulative mutation platform. So it accumulates mutation over time. So we have collaborated with Reza Kapoor and George Church to use the homing CRISPR mouse coupled with NSC-seq, where we can basically capture the guide RNAs and read out the sequence. So we can read out the mutated sequence as well as the mRNA in this particular platform. So this is just some validation. So with regards to time, we would expect the mutation density to increase over time, like this model here. And in this is what we saw from organoids as well as from the mouse. There's a linear increase in the accumulation of mutation density. And then we also can look at lineage reconstruction. So we have these control passage experiment where we have the ground truth of how and when we split the cultures. And then we can reconstruct the parental lineage based upon NSC-seq using both DNA as well as guide RNA. Okay? So here we are barcoding normal. This is just to remind me that we're barcoding normal cells now. So we can answer a lot of questions in developmental biology. But what we're interested in is couple this barcoding approach to tumor initiation. And the simple question that we want to answer is that we're barcoding normal cells, right? So if we generate a tumor in this mouse, would the tumors be comprised of a single barcode from these normal cells or from multiple barcodes? So we're talking about monoclonal versus polyclonal tumor initiation. So this is, again, a control. If you look at lymphocytes within the PBMC, we know that for the most part, excluding clonal hematopoiesis, those will be polyclonal. And we do see that there's multiple, like these little blue squares here. Whereas if we look at tumor-infiltrating lymphocytes, what we now see is the signs of clonal expansion, right? So these squares get bigger. So now if we look at the tumor cells, if the tumor cells came from a single cell or single clone, single normal clone, we would see one giant square of the mutations that are shared with the normal cells. But what we instead saw was that there are the tumor is comprised of multiple clones that are derived from multiple clones from the normal tissue. And these clones have very different mutational profiles to each other. Okay, so that would imply a polyclonal tumor initiation process. So that's one type of data. We also have orthogonal data. So here, what we do is we're trying to count the number of driver mutations. So in the context of colon cancer, the driver mutation, the initiating event is driven by 2 biallelic loss of APC. So we should see at most 2 mutations in APC within a tumor if the tumor is initiated from a single cell, given that the 3rd mutation should not be selected for. But what we saw was that there are multiple tumors with more than 2 APC mutations, implying that these tumors arose from multiple normal clones. So because we're doing this mutation counting exercise, we can easily apply this to human. So we go back to our H10 cohort, as well as a larger teacups cohort with hundreds of samples. And what we do here is simply count the number of APC mutations. And approximately 20% of the polyps, the precancers, have more than 2 APC mutations. Whereas if we do the same exercise in cancer, greater than 95% of the cancers that we look at only have 1 APC mutations. Okay, so that again is one piece of data. We want to look at orthogonal data. So here we took advantage of X-linked inactivation. So in a biological male, we have 1 X chromosome. So if we look at the transcripts from the X chromosome, that all cells, a majority of cells, should have the same SNP in X. Whereas in the females, we can take advantage of this X-linked inactivation because so if we look at any SNP in the X-linked transcripts, then it's expected to decrease, given that it's a kind of a mosaic. And what we want to see if this mosaic sort of carries into the precancers. Okay, so first we did a simulation. We took male polyps, which are thought to be clonal, and then we mixed them with another male polyp, so simulating polyclonality, and we do indeed see a drop in clonal SNPs in X. So here it allows us to sort of set a threshold of monoclonality and versus polyclonality, where we can then apply to female polyps now with X-linked inactivation. And then indeed there are 30% of these polyps that are fall below this threshold and are thought to be polyancestral. So this shows that it's possible for tumors at the tumor initiation stage to be initiated by multiple normal cells, which validates some of the mouse work done by Richard Holberg a number of years ago, highly controversial, in terms of polyclonal initiation, because the knowledge in the field was that cancers come from a single cell. So, you know, the state of the system, as we talked about, right? Cells and the niches can now be assessed quite easily by single cell and temporal and spatial technologies, right? But what we're missing is this temporal component a lot of the times, right? Because we're taking these snapshots. But you can actually use genetics, which are relatively unidirectional, to act as a temporal scaffold. And then you can think about using these mutational barcoding temporal strategies that on non-tumor cells, like stromal and immune cells, to depict the dynamics, origins, and clonality during precancer initiation and progression. And people are actually starting to do that, right? So there is a hot off the press publication in the New England Journal about how tumor infiltrating clonal hematopoiesis were predisposed to tumor progression, et cetera, right? So the implication of this is that, you know, with monofits, the monoclonal polyp seems to be under more selective pressure than polyclonal, right? And we saw that the monoclonal polyps have expressed higher stemness, higher Wnt signaling, and when you look at the tumor microenvironment, also a higher exhausted T cell signature, somewhere in between the polyclonal as well as the colorectal cancers. So it seems like the monoclonal polyps are in the process of progressing into malignancy. So this is kind of, you know, a significant development, because the current state of the art of assessing risk of any polyp is still based on the size of the polyp, or any precancer, it's based on the size of the precancer. And perhaps we can look at other genetic measures to determine risk, right? So there are a couple other studies that sort of work in this area. So Doug Winton, you can see clearly that you can label individual crypts with different colors, and then the polyps will have different colors because monoclonal. And you know, this is another approach using DNA to sort of look at this tumor initiation process. And I just want to leave this picture with you, right? Tumor initiation is not a stepwise process, right? It is actually an evolutionary process controlled by many, many bottlenecks. So at the end, I want to go over some negative data with you, and I think it will lead to some discussion with the synthetic biologists, right? So as we all know, NIH has made some policy changes, right? With regards to the reduced use of mouse models and focus more on human-based models for research. And I myself, before this initiative was suggested, was trying to get away from mouse, to convert my mouse lab into a human organoid, organ on a chip type of lab, right? So let's talk about, you know, the deficiencies, or, you know, of the weaknesses of current models of tumor initiation, okay? There are many, because of mouse, there are many differences between mouse and human, for example, microbiome differences. We can talk about all of that. But with regards to tumor initiation, again, right, cancer is sort of disease in aging. And basically, this process occurs early in life and takes decades for it to manifest, right? But for mouse models, what we want to do is induce the tumors in a number of days, right? And we're doing this through driver mutations like APC. So I would argue, right, at best, right, the models that we're looking at for tumor initiation are models of these genetic syndromes that lead to tumorigenesis, like FAP, like Lynch syndrome, etc. Not completely the same as sporadic tumorigenesis, which is what we claim that we want to study, right? So, you know, cancer is a disease of the aging host, perhaps the aging host microenvironment. So an example of which is, you know, the accumulation of senescent fibroblasts, right? That sort of over age would create a tumor microenvironment conducive of converting these dormant precancer normal cells into precancer and then cancer, for example. We're missing that, right? So a mouse is not a human. So I've heard that many, many, many times. But then the question is, do in vitro models sufficiently recapitulate the tumor microenvironment for studying spatial biology, right? Which is, the topic of what we want to discuss today. So let's take the simplest system, human precancer organoids, and look into this. So this is unpublished work, maybe unpublishable work by my student Monica Brown. Okay. So we know that we have to culture these human-derived material in a variety of different media, organoid media, right? So I just want to make a comment that the media that we culture all types of different organoids, from gut to brain to liver to any type of organoids, originate from the Hans Clever's Sato protocol, which was optimized in the gut through picking out developmental pathways, right? So we're modifying those media by a little bit to optimize those media for growth in these different tissues. So it's optimized for growth. But the in vivo microenvironment, I would argue, may not be optimized for growth, right? So if we actually profile single-cell RNA-seq of adenoma organoids grown in OGM, it's very different. I mean, people know this, right? This is not a surprise. Very different from in vivo tissues. But what is more of a surprise is that if we culture the same tissue as organoids in different well-known media conditions, they are also very different from each other. Okay, so that's number one. So the organoid phenotypes, at least from a transcription perspective, exquisitely sensitive to culturing conditions. And then what do those phenotypes look like? Well, there are unexpected changes to cellular identity. So, these are, colonic, precancers, and they adopt small intestinal phenotype as well as a fetal phenotype, right? So it's very, very different from what we, see in tissue. Now, more importantly, and more of a problem is that, the closer you are to the original tissue in terms of passages, the closer those organoids, I wouldn't say resemble the original tissue, but they are more consistent, right? So the phenotype, the transcription profile, also would drift as a function of passages, okay? From both the precancer perspective as well as the normal perspective, okay? So that's another point. The third point is that, remember, when I show the tumor progression, it is not a linear progression spectrum. It is, you have to think about it from an evolutionary perspective, right? There are, times of equilibriums and times of selection. And we have, detailed genetic profiles of the tissue. But then when we look at the organoids, so serrated lesions, they don't have a lot of stem cell capacity, but we can grow them out. But what we grew out, there's serrated lesion organoids, the genetic profile is completely different from the tissue. Actually, when you compare that to sort of the known oncogenes, also very different. So this begs the question of what we are actually growing out in these organoid systems, right? So the food for thought is, okay, we have to examine the in vitro models, right? From a tumor evolutionary point of view, foundry effects. What are we actually growing out from tissues initially? And then how does that mirror phenotype, right? Secondly, when we talk about the tumor microenvironment, it's highly complex, right? So it is very highly likely that one media condition is only going to be conducive to a couple of cell types, and not the complex tumor microenvironment. Thirdly, I think there's a lot of work now done on sort of air-liquid interface, explant cultures, et cetera, right? Where experiments are done sort of, low passage tissues. But then that speaks to the reproducibility of the results. Can you repeat that over time, right? Because, the explants, you can only do that once, right? And then also a larger question of, I keep emphasizing this. We use the models with the data, but specifically to answer a biological question. But this is very different from a different synthetic biology engineering question where we're trying to build models that would recapitulate in vivo physiology, right? So that would replace the mouse models eventually, right? I think we're some ways off, but I think this will be up for discussion. So with that, I would like to stop here and maybe we can have some discussion. Thank you.
Thank you, Ken, for that very thought-provoking presentation. You left us with a lot to think about at the end and hopefully to start a conversation. So we have about 8 minutes for questions now. So maybe I'll start off with a question from the Miro board. So I'm going to interpret here a little bit, so please forgive me. But one of the questions was about the issue of cooperativity versus competition in these polyclonal initiation events. And so can you, for example, look at the statistics of the clonality and try to ascertain whether these very early events are somehow selected for in a positive or negative way?
Right. So this question is a great question. I think the thing that we have to think about is that the tumor initiation as well as the tumor progression process, it takes over decades, right? So basically, I think there would be different time periods or epochs of selection as well as cooperation. With regards to polyclonality, right? There's definitely collaboration or synergy because so basically there would be what we call mutations that are weak. And actually, I would argue most mutations, it's easier to break things than to build things, right? So most mutations would confer less fitness to the cells, right? So polyclonality is a way by which cooperation between the different clones would allow these weaker mutations to persist. So the mechanism by which this is done, actually, there are many, one of which is actually through secreted factors, like for example, NODO, which is a negative Wnt regulator, will allow for certain clones to persist in a tumor microenvironment. So that is at the very early tumor initiation stage that you allow some of the weaker mutations to compete in this environment of cell competition. But I think when you transition now to precancer to cancer, then there would be one bad clone that evolved to accumulate additional mutations that would, so we don't really study that part, but our hypothesis is that would outcompete others due to resource availability, like using certain metabolites, like glutamine, et cetera, with KRAS, for example, right? Because if you have KRAS, then it converts polyclonality to monoclonality. So I think, so the answer is cooperation and competition, they both exist, but I think they exist in temporally discordant periods of time.
Excellent, thanks. We have another couple questions on the Miro board that pertain somewhat to nomenclature, and maybe we can get your thoughts on that. So one was, I'm going to try to reframe things here. One was that it seems that what we talk about precancer could be so flexible that essentially any tissue state could be considered a precancer, given the fact that there's mutations all over the place. So can we so the question was, can we clearly define what is not a precancerous state? And then maybe if you could also at the same time comment on what might be the differences between a benign tumor and a precancer.
Wow. So, I think in the field, I think people, there was a pre-cancer think tank that happened in 2023. There's a recent publication from that think tank to discuss precisely that, what is a pre-cancer, right? But I think the majority of the people, think about pre-cancer as a lesion, morphologic lesion, right? And I think that's the official definition. I think a way more interesting question, as the person asked, is what is defined as not a precancer, right? And I think the definition of precancer would be cellular states that would eventually lead to cancer, right? So, I mean, this gets very philosophical because you can imagine that given infinite amount of time, all cells can become precancer, right? But I think a related point that I would want to make, right, which may not directly address the question, is the order of mutations. Okay, so with the Vogelstein model, right, you look at APC and then KRAS because APC occurs more, right? But you have to think about KRAS would exist in normal tissues, right, a lot, but they are not found as much in cancer because the APC mutations are necessary to drive the cancer, where the KRAS not. But I think there would be differences in the cancers that are APC and then KRAS that result from that versus the cancers that arise from KRAS and then APC. And I think there's actually work in progress now to define those different subtypes. And then I think the second question, if I remember right, what is a benign lesion versus a precancer, right? So I think a precancer encompasses both benign precancer and malignant precancer. And again, you can argue a benign lesion that would never, I don't never say never, right, but never progress to cancer would not be defined as a precancer. I hope. Again, I'm not the authority on this, so I hope that just shed some light on this discussion.
Yeah, the nomenclature questions are tough for sure. But there's also a couple questions in here about, I think, this very important point that you brought up about media and/or microenvironment in vitro for how we go about trying to model
Right.
These tumors. And so maybe to add my sort of own twist on these questions, what is your thinking in terms of the directions that we need to go to build in vitro models where we get closer and closer to something that looks in vivo? How should we be thinking?
Right. I think the field, right now, the state of the art is, again, I don't want to be too controversial here, right? But in terms of developed media for differentiation to one cell type versus another, it's the starting point is always through the original media recipe, right? And then we're modifying the original media recipe to include different, different components that are informed by mechanistic studies. But the reason I brought that point out here is because I think there is significant opportunity for systems biologists and synthetic biologists to collaborate in this aspect, to really start from first principle to inform the tumor microenvironment from human systems level studies, looking at cell interactions as well as the cytokine ECM microenvironment to build better models that hopefully we can get to recapitulate the tumor microenvironment. What I'm trying to say is maybe we should start from first principle, right? Because the cancer is very different from normal tissues, and precancers are very different from normal tissues, right? And I think it will serve us well to sort of start from first principle and tailor each of these conditions to try to recapitulate the TME.
Great, thanks. There's also a few questions on here about the immune system, and one of these questions pertains to the mouse model in particular, asking, in some of these genetically engineered mouse models, how do we think about the antigens that are being recognized by the immune system, and how might those be relevant to what's happening in humans?
We're working on that right now, right? So the question is, we get that question a lot, right? No neoantigens. There are some, right? But then they're very low. So what is the immune system reacting to? So actually, if you look at the fetal reversion phenotype, so one aspect of during embryonic stages, right? So is that there's epigenetic regulation of the genome that's large scale during embryonic development. And what we saw was that some of these mechanisms are active in the precancerous lesions in human. So we are now very focused on these epigenetic changes, focused on antigen that are re-expressed, like transposable elements that could be immunogenic. So we have some evidence now that this is sort of indeed the case at the first step. This embryonic reversion cellular plasticity stage, that's one of the programs that is activated, hijacked by these precancers that sort of allow this immune regulation. We have now some mouse models also actually that are recapitulating serrated tumorigenesis. These are all from damage, and then we're looking at this aspect.
Great, Ken, that was really fantastic and a great conversation. Thank you everybody for posting your questions. And remember to keep the questions, challenges, and opportunities coming as we go through the session today. So we're gonna adjourn for a quick break, and let's all plan to be back here promptly at 1:20 to keep going.
From University of Pennsylvania, and really excited to moderate our first conversation here. What's already been a very thought-provoking symposium. So the way this will work, it'll be different from a normal conference. We're going to have a set of 3 different conversations. Conversations where we will have 2 speakers speak for 10 minutes each, very short talks, and there will not be questions at the end of each talk. We welcome you to put your questions in the Miro board like you did for the first presentation, and then we'll have after the 2 talks we'll have a discussion between the 2 speakers, and that will be 20 minutes. We may pull in some questions during that discussion depending on time, but after we've had 3 of these conversations, so 3 sets of 2, we will then have breakout rooms where that'll be a chance, more of a chance for everyone to interact with the speakers and the topics. More verbally versus just on the Miro board. So, but because of the short time frame, I really want to get started to not take away too much time. So I'm going to welcome our first speaker, who is Arthur Lander, who's the Donald Bren Professor of Developmental and Cell Biology at UC Irvine, and also the director of the Center for Complex Biological Systems. And he will tell us today about modeling how cells can escape population-level controls on proliferation. Take it away, Arthur. Arthur's muted.
Okay, how about now?
Yes, we hear you loud and clear.
All right, let's, can you see my screen?
Yes, yes, it's perfect.
There we go. All right, so thanks for the introduction. Actually, I think it's really great coming right after Ken's beautiful talk, because what I'm going to talk about is how explicit mathematical models can produce exactly the kinds of slow polyclonal transitions that Ken was alluding to. So let me begin just by reminding us that in biology, we always like to portray causality in terms of deterministic linear pathways, like this EGF receptor pathway, A goes to B, goes to C. And even in the famous Vogelstein way of approaching cancer initiation, it's also that same sort of linear causal pathway. Although one difference is that each causal step is a stochastic step, but nevertheless, the idea is that mutations, even though they arise stochastically and within one cell lineage, then act deterministically. And I'm going to argue that this way of thinking about causality actually holds us back a great deal. And it would be advantageous for it to let go of this bias quite a bit. So what other way is there to think about causality? And within systems biology, we often take a dynamical systems approach. We think about whole systems and how their dynamics can be attracted to particular final states. So in this easy differential equation system, you can see no matter where you start in the state space, the system as a whole always progresses to one particular steady state solution. So that's called an attractor state system, and we can often represent the same system using this potential well formalization, where the idea is that the system is kind of like a ball circling around a well and attracted to one particular point at the bottom of that. So in this view of causality, causation is viewed as kind of movement towards some place the system has to go. Stochastic processes push things along, but you don't really have to pay attention to all the details of how something gets somewhere. And that's a different way of thinking about causation. Now, one of the reasons why I think it's so important to take that attractor-based approach to thinking about the control of proliferation is you can't even achieve simple things like homeostasis without having an attractor system. Just to illustrate that, I have this picture here of how we typically think of stem cells behaving, giving rise to two cells, which then with a 50% probability can become stem cells, or 50% probability become terminal cells. And we know from a lot of experimental studies that these really are probabilistic decisions that are made at some point during the differentiation event. And so what this produces is a system that on average will keep the number of stem cells constant, and on average will produce terminal cells at a constant rate. But if you actually model it stochastically, what you find out is that the individuals never behave like the average, and they wander off into either very huge or very small states, and the coefficient of variation between them grows without bound. And the reason is that fine-tuning can control only the average behavior, but the individual variation requires more control. Essentially, just to have robust homeostasis, you have to have some kind of attractor system. So how's that done? So there are a number of different ways, and I don't have time to talk about them all, but one is called renewal control, where the cells at the end of the lineage produce some diffusible molecules that simply feed back and affect not the rate at which the stem cell divides, but the probability that its offspring will become terminal cells as opposed to staying stem cells. And when you do that, you turn this crazy system into a very nicely, tightly controlled system, because you've created a single attractor state at a particular steady state value that the whole system is drawn to. This is called integral negative feedback. It's a common motif in systems biology. It gives you global stability, this phenomenon of robust perfect adaptation where nothing matters other than the things within the feedback loop, and it can reject all kinds of noise or disturbances. Essentially, renewal feedback just creates a dynamical attractor system where the number of cells is always drawn to a particular point, depending upon whether terminal cells turn over or whether they don't. So as I mentioned, attractor systems can be great at suppressing noise, but when you have multiple attractors, you can have discrete transitions that are driven by noise. So a classic multiple attractor system is the toggle switch system that's used very commonly in driving cell differentiation. When you look at it deterministically, this produces a system depending on where you start in terms of who's stronger, A or B. You either go to all B or all A. And when you do that in the presence of a bit of noise, you still end up going to a particular state. But if you have a little bit more noise, you get these rare events where occasionally something that ought to have gone to A occasionally wanders off into B. That raises an interesting philosophical question. What's causing this rare event here? At some level, it's the noise. But on the other hand, the noise is almost never doing that. So on some level, it's the attractor landscape. Well, I guess it's both. So we kind of ran into this particular problem when Micah Caldwell in the lab was using agent-based modeling of this renewal control process in order to see how it changed when you actually modeled it in space, rather than in a simple non-spatial model that I showed you before. And so he built an agent-based model in which terminal cells produced a gradient of material that then fed back on the dividing cell shown in orange. And in the agent-based model, things kind of move out as a lattice. And so he simulated this, many thousands of times, and the vast majority of times, these would behave just like the non-spatial model of renewal control. They'd all come to rest at a particular final state. And when he tallied them up, they fit a nice log normal distribution, which is what theory predicted. But every so often, he'd see one outlier that looked like this, that would just keep growing until the computer had to stop because it was taking too much memory to simulate, but clearly would have gone on growing forever. This was also the case when he built somewhat more sophisticated models where he allowed the terminal cells to turn over. The same thing, most of the solutions were in one narrow domain. Occasional ones would be way outside and would keep going as long as he could simulate. Now, could he explain that kind of behavior? So it turns out space creates a cryptic positive feedback loop in this system. Essentially, if you get a collection of stem cells that are in one part of space and they grow even bigger, that pushes them farther away from the source of the negative feedback coming from terminal cells. That thereby increases the local renewal probability, which increases the probability that they'll go on to another stage where they push those terminal cells even farther away. And you can see in these cases that explode, you have these big blocks of stem cells that are growing faster than terminal cells can fill in. And essentially the whole system has gone out of control. In effect, we turned a single attractor system, which had one nice steady state, to a multiple attractor system, which has the possibility of wandering off to infinity. Deterministically, that will never happen if you start from a low state, but stochastically it will. You will occasionally have trajectories that collectively escape control, right? So there's no individual cell here that's driving this, the whole population's doing it. And this collective escape from control acts like a simple constant probability stochastic step, just like a mutation, right? So we can simulate this and show it has this on a log plot, a linear decline in survival. So that's a typical single hit kinetics. And we wondered whether this kind of collective escape could account for random steps on the path to cancer that simply don't seem to be caused by mutations. And this is an example from some work we do in collaboration with Anand Ganesan's lab and Karen Shao, who did these beautiful studies on initiating melanomas at very low probabilities with just BRAF or just BRAF plus one allele of PTEN. And these behave like single hit kinetic stochastic transitions in these mice. But when they're sequenced, there are no sort of causal mutations to produce these tumors beyond the ones that were engineered here seem to put them in, even though they're rare and seem to be occurring according to a stochastic step. So how can we test this idea? Well, one of the differences about these kinds of transitions is they should be reversible, right? Any kind of collective transition that's just based on the circumstances of the cells presumably could be reversed if you change those circumstances. And we note that after sort of potentially curative treatments for various cancers, cancers often recur and sometimes very much later. The traditional model is that, well, there's some surviving cancer cells and they're still cancer cells. So they somehow regrow, even though they might be doing it very slowly. And that's a deterministic way of thinking about the cause of a recurrence. But the alternative possibility is that in response to treatment, cancer cells revert back to a state on the other side of this separatrix. And now they sit there waiting for the opportunity to then collectively re-escape stochastically as opposed to deterministically. One way we can distinguish between those models is actually build models of stochastic versus deterministic escape and apply those models to all kinds of data that are in the literature on rates of cancer recurrence after potentially curative treatments and see how well they fit. And while we can't rule in or out any particular model, what we can say is that for the vast majority of the cancers we've looked at, the stochastic model of cancer recurrence is at least as, if not often, a better fit to the data than any of the deterministic models we've looked at. Now, more generally, this idea of stochastic escape from growth control, which I've illustrated using renewal control, actually is a very general phenomenon. It will arise in space whenever growth rises faster than feedback. So here, for an example, this illustration is of something growing as a sphere, but being removed, for example, by the immune system at the periphery, basically attacked from the outside. And what you see is as long as it's small enough, removal can exceed growth. But once it gets beyond a certain tipping point, growth will always exceed removal. And you can show that even under conditions that deterministically should always be removed, occasionally you'll see trajectories that wander off forever, again, in this simulation. So the implications are, because a potential for stochastic escape from control always exists in proliferative systems in space, it is not necessary to posit a deterministic cause for every step in cancer. It also means that cancer mutations don't have to work by making cells grow or making them grow uncontrollably, which is how the sort of typical hallmarks approach to cancer makes us think. Cancer-causing mutations could simply be ones that alter the attractor landscape to make the probability of stochastic escape a little bit higher than it was before. And so I'll finish by thanking the folks in my lab, particularly Micah and Anand and Karen, for the work I talked about. Thank you very much.
Thank you, Arthur. That was fascinating and a beautiful talk. Look forward to discussing it in about 10 minutes in our conversation. But for now, I would like to move on to our next speaker, who is Mo Khalil from Boston University. So if we could pull up Mo's...
Thanks, Lucas. Let me see if I can.
Just as Mo gets the slides up, just a quick introduction. Mo is an a professor in biomedical engineering at Boston University, also the associate director of the Biological Design Center, and he will tell us today about synthetic technologies to study signaling and epigenetics in cancer. Thanks, Mo.
Thanks, Lucas. Let me see if I can get this. Oh, it's not letting me share. Uh-oh.
Want us to jump in and share for you?
Sure.
Okay, Joshua, if you wouldn't mind pulling up Mo's slides, you can next
I think
slide.
I just don't have my settings right. Yeah, that would be great if you can. I think the slides
WebEx
are pretty
likes
much
to
the
trick
same.
people up.
Yeah. Thanks, Michelle. Oh, look at that. Perfect. Thank you so much. And I'll just hit next, or when we need to advance. Thanks, Lucas, for the introduction. Thanks to the organizers for putting together this wonderful workshop that brings together synthetic biologists, systems biologists, and cancer biologists. Just to be clear, I'm a synthetic biologist and just kind of very shallowly think about cancer. So let me just give you a little introduction first. We develop tools of synthetic biology that allow us to construct and dissect genetic circuits in living cells. Just advance here. I think there's a lot of transformative potential in this approach. We basically use this, to turn biochemical reconstitution into a genetics problem, to study mechanisms and principles. In the middle there, we use synthetic circuits to enact precise control of therapeutic cell function. And on the right, we even deploy genetic circuits in the context of laboratory evolution experiments to sort of steer evolution to evolve interesting proteins or to study the process of adaptation. Now, all of these, I think, are useful in the context of studying cancer biology. Today, we're thinking about cancer initiation. And I'll tell you a little bit about two unpublished stories around this. Advance, please. But before I do that, maybe just a quick note about some work we've done in that kind of middle category to develop synthetic biology tools to enhance cancer cell therapy, such as CAR T-cell. I think you'll hear about this later in the workshop. I won't talk about it here just to say we've developed a number of really, I think, exciting tools that help us basically control the function of CAR T cells and their anti-tumor functions. Two of those tools are sort of depicted here. The Synzifter transcription regulator platform that allows us to build genetic circuits that can be controlled by FDA-approved small molecules, and work led by Cole Roybal's lab with these sort of synthetic receptor systems, that allow basically T cells to activate gene expression programs in response to tumor antigens. Okay, so next slide. So now into the meat of the talk. I'll talk about signaling and I'll talk about epigenetics. And first, signaling, right? So mutations that alter signaling, of course, are drivers of oncogenesis. Next slide. However, even within a single signaling pathway, such as the PI3 kinase pathway, not all activating mutations are the same, of course, as we know. Here's a Kaplan-Meier curve for patients with breast cancer. And what we're plotting here, of course, is the overall survival in patients harboring various mutations that all, by the way, lead to increased PI3K AKT signaling. So even despite that, you can see that the median survival varies substantially, right? And this discrepancy suggests that the PI3 kinase axis activating mutations look and behave quite differently, I think raising a lot of intriguing questions. Next slide. So we're actually quite inspired by really seminal work by our organizer, Lucas, who used optogenetic tools and biosensors to suggest that altered signaling dynamics may contribute to tumor progression, right? And they showed, for example, in this beautiful study a number of years ago, that, for example, common mutations like BRAF G469A slows the kinetics of the pathway, thus basically forcing cells to misinterpret signals and driving oncogenic proliferative responses. So we wondered if the distinct phenotypes seen in the PI3K axis could be explained similarly by differential changes. Next slide. Okay, next slide. So we examined two common mutations, the ERBB2 amplification and one of the PI3 mutations that leads to constitutive activation. What we found is expressing either of these lesions in non-tumorigenic MCF10A cells, we find that ERBB2 amplification drives proliferative response, PI3 kinase lesion drives a more EMT-like response. And if you click, in addition to that, these two lesions drive very different signaling dynamics in response to a PI3K agonist. So you can see it right here, for example, the ERBB2 amplification has a significantly prolonged or intermediate response, whereas the PI3K mutation shows this kind of sustained sort of constitutive activity. Next slide. So given these differential dynamic responses, much like Lucas, we wondered if mutation-specific rewiring of this signaling axis could contribute to the distinct phenotypes that we observed, which I didn't show you data for, of course. So Keith, who led this study, set out to design and build basically an optogenetic PI3K platform that enables us to sort of orthogonally control the intracellular PI3K activity and deliver mutation-like signals to these non-tumorigenic cells, okay? And so if you click, we developed this optogenetic tool based on the eyelid nano platform, which has some of the fastest on-off kinetics. The data on the bottom there is just showing you that we can basically activate or deactivate the PI3K pathway with really rapid kinetics. Okay, and just in the interest of time, I won't get into sort of the details of the engineering here. Next slide. Okay, so using this optogenetic synthetic biology tool, we could deliver these mutation-like signals to, again, wild-type MCF10A cells. These are non-tumorigenic cells. And interestingly, what we found is we could, when we were able to demonstrate that sustained PI3K stimulation was able to phenocopy the EMT-like changes associated with the mutation. That's kind of on the two plots on the left. But interestingly, stimulation of MCF10A cells with the intermediate duration stimulus, that mimics the ERBB2 amplification, was unable to phenocopy the proliferative response. And so this suggests that, in some cases, these signaling dynamics are sufficient to drive the phenotypes, like in the EMT-like, whereas in others it's insufficient, meaning there's probably additional signaling changes that are needed. Okay, so if you click. All right, so there's a lot of exciting next steps to tackle here. I won't read this out loud. We'll just kind of go to the next part of the talk if that's okay. Second, click to the next one. Okay, so I'll be very brief with the second half. The second area I want to touch on is about epigenetics, and these are sort of stable changes to gene expression that can't be explained by alterations to DNA sequence. Most famously, through these chromatin changes in cells. And of course, we've known for a long time that chromatin-based epigenetic regulation is fundamental to development. The famous Waddington landscape, of course, to how organisms adapt, and very recently, implicated in cancers, of course. And of course, while we think about cancer as arising from sort of genetic mutations, there's now increasing evidence that epigenetic processes play a really important role in both transformation as well as the persistence of cancer states. In this really beautiful example from last year, in fact, the authors showed that, in fact, just a transient disruption of heterochromatin machinery in Drosophila can induce an effectively irreversible and permanent cancer cell fate. I think this is a really beautiful example, kind of spinning the paradigm on its head to some degree. Okay, so click, please. And I think those of you that know me know that for many years my lab has taken this kind of synthetic biology approach to define the basic requirements and principles of epigenetic inheritance. How do we get these heritable gene expression states? And this has been fruitful through a synthetic approach because these processes are highly dynamic and often involve redundancy. So it's really not good to study from the context of sort of genetic knockouts, let's say. Okay. Next slide, please. All right, so recently we've been focused on heterochromatin, this epigenetically inherited chromatin domain. And if you look at the tree of life, what you see really is that heterochromatin, possesses multiple different epigenetic modifications in addition to the hallmark H3K9 methylation, right? And in addition to that, it's been observed that these modifications are coupled to one another in a phenomenon that we like to refer to as epigenetic crosstalk, right? And in fact, there's a really great example of this in the context of cancer, and that's this protein UHRF1, which reads both H3K9 methylation and DNA methylation, kind of coupling these two together. Interestingly, this molecule is a biomarker and an interesting therapeutic target in cancer because when it gets upregulated, this contributes to silencing of tumor suppressor genes, right? So we've been asking a lot of interesting questions around how we can use synthetic biology to study this enigmatic process of coupling and crosstalk in epigenetics. If you just click, I'll show you just one slide on this. And actually just click through if you don't mind, just in the interest of time. One more. There we go. Okay, so in this work led by Colin Coombs, we've taken this two-pronged approach where we've investigated these principles of epigenetic crosstalk, both through mathematical modeling and synthetic biology. With modeling, what we've done is basically develop these minimal stochastic models of chromatin modification. And this allows us to look at the changes of these chromatin modifications in space and time. And without getting into tremendous details, this has taught us some really interesting things. For example, the ability of crosstalk between modifications to support the maintenance of a confined or localized chromatin state. That's what you see there on the top right. That's memory, but without unregulated spreading, okay? In addition to that, it's taught us the advantages of maybe using distinct modifications and coupling them to one another, such as histone methylation and DNA methylation. In addition to that, what Colin has done is coupled this with experiments. We've been using actually the model organism fission yeast, S. pombe, which is really this great model for exploring the basic principles of heterochromatin because it has a simplified heterochromatin system. It's very attractive to us because pombe and actually many yeasts just lack DNA methylation entirely. So what we have done is basically built DNA methylation into the system and coupled it to the native K9 methylation, layering crosstalk on top. We have found some really interesting things. We've discovered regimes that drive totally different levels of epigenetic memory, from very transient memory to highly durable, over 60 generations of memory. I kind of think of this as a two-factor authentication system, whereby coupling between different modifications, can allow organisms to build and protect memory this way. All right. So next slide. I'll kind of sum up there. Yeah, great. This is just my group. The last thing I'll just say is I think, we all can appreciate that it's difficult to sort of discriminate among the various contributions to tumorigenesis, whether that's genetic, environmental, or even these kind of cell intrinsic epigenetic processes. And sort of hopefully what I've shown you here, and maybe the discussion will explore this a little more, is that synthetic biology tools, because of their ability to enable precision perturbations and reconstitution, can maybe help us distinguish between these highly sort of interconnected, if you will, contributions. Okay, and I'll stop there and give it back to Lucas. Thank you so much.
Thank you, Mo. That was fantastic. Yeah, actually a really wonderful combination of the first three talks. So for the next 10 minutes, we'll discuss a little bit on everything we just heard, and then we'll move on to the next conversation. So we have Mo, we have Arthur, and myself, and I think we're front and center now. I'm not sure what we're seeing, but let's assume we are. So let me just start with a couple of questions. Maybe first I'll go to Arthur. So I think that one of the whole points of this symposium was that we hear some of the big challenges in important areas of cancer biology, and then try to bridge new ways to look at this both from a theoretical and an experimental perspective. And so, Arthur, my question to you is, with this really elegant model of how of tumor initiation, what I guess to what extent has this been tested experimentally in either in culture or even in more complex models, and what's missing, right? So what from either what you just heard or in general in synthetic biology would be the sort of most I guess fruitful thing to test next, to sort of test these models?
Yeah, that's a really good question. One of the the difficulties with having a high level theory is often there's many different ways to implement it, right? And so it doesn't tell you exactly what molecules to go after. I mean, certainly one level of testing is to look at animal models of tumor initiation and see if you can see evidence for this sort of collective polyclonal causality. And I alluded to some work we've done with melanoma that supports that. There's some great work being done at our institution by Rick Van Etten on chronic myelogenous leukemia that absolutely shows completely polyclonal initiation of that cancer after getting the initiating mutation. So I think we're going to see more and more examples of this, where it's not that the mutations are unimportant, it's just that there's additional steps that involve collective behavior of the cells that harbor those mutations. But to get at the real mechanistic level, you need to know what are the circuits that are creating these attractor states. And as I tried to allude to, they can be things like simple negative feedback loops involving diffusible factors, but I really think that's only the tip of the iceberg. And as I mentioned, just interactions between immune cells and normal tissues can similarly create the same sort of circumstance. From an engineering perspective, these are all cases of what engineers call declining gain feedback control systems. And they all have that same fragility. And so I think we need to both do more theory to figure out what are the range of possible ways you could get these kinds of declining gain feedback systems, and then find a way to really nail the molecular mechanisms that are underlying those different network configurations.
Yeah. And in your estimation, I guess, is the most fruitful place to look kind of how signaling, kind of perturbed signaling like Mo showed, can reshape these attractor landscapes, or is it epigenetic regulation, or is it all of the above, or some combination?
Yeah, I mean, epigenetic regulation is another one of these attractor building systems, right? I mean, these are all attractor states that when we talk about cell state being controlled by epigenetic regulation. Unfortunately, I think partly what this is going to force us to do is to better understand the networks, the intercellular networks that operate in normal tissues. Because if we don't understand those, we're not going to understand how you escape from those. So I think we're going to have to do not just looking at tumor models, but also models of normal tissues even before they become precancer.
That's right. So actually, and Mo, I guess that feeds in nicely to you. So you showed us some really great perturbation tools on the signaling and epigenetic landscape, but also you have a lot of work in cell engineering as well, as you alluded to at the beginning. To what extent are people already starting to look here to say, you know, think for the most part, people think of engineering T cells for, you know, killing cancer cells and so on, but they can also go into microenvironments and modulate, you know, they can screen any number of things that you design. Those would seem to be really great tools for these types of questions, but I don't know. Is this something that's actively underway, or what's the potential there?
Yeah, absolutely, Lucas. It's a great question, and yeah, I would say, you know, historically, at least among synthetic biologists, and certainly in our lab, right, the dogma has been, you know, thinking about molecular circuits and individual cells, and the CAR T or the T cell happens to be a really dominant and kind of powerful example of this, but, you know, limited in that way, right, as you said. And I think that the future for many of these tools will be to sort of deploy them in the context of, as you said, right, either tissue engineered models, which is something that we're thinking a lot about with, for example, folks like Chris Chen and others, organoid models, or, you know, other types of more complex systems where you can incorporate cell-cell interactions and some form of, like, you know, multicellular systems. Because as Arthur beautifully, spoke to, many of the sort of phenomena and the feedback loops, the cryptic feedback loops, if you will, really arise from those types of interactions rather than just at the level of our classical thinking of molecular feedback loops and molecular circuits. And so we are very excited about that. I mean, of course, there's, you know, there are very sort of technical, you know, considerations there of sort of how it is that you perturb or engineer certain cells and layer those on to sort of some of these more complicated, you know, tissue-like models. But those are, you know, technology-related problems that I think are solvable.
I guess one maybe open question to either of you is, what sort of, did you sort of learn anything from each other's talks that's sort of on the very more theoretical side and the very kind of applied perturbative side? Kind of any interesting connections or links that maybe you hadn't had before?
Yeah, well, certainly, you know, one thing, and this would be, you know, Arthur, is really great to hear your talk. I mean, the sort of one thing that just jumped out at me, and, you know, immediately was just, you know, the progression. You're sort of initiating cancers in the absence of genetic mutations, right? Or the progression of, you know, the cancer development in the absence of mutations. And this, of course, really resonated with me because as someone who studied epigenetic processes for, you know, some time now, and seeing some of this new work that sort of suggests that you can even initiate cancer to these irreversible cancer cell fates in the absence of mutation, I think was really exciting for me.
Yeah, and for me, it was just exciting to hear how much we can build into the engineered cells that we can make, and kind of made me hopeful that it might be possible to actually create entirely in vitro systems that show the entire process of both robust regulation of proliferation and then occasional stochastic escape. Because if we could see that in vitro, that would just give us tremendous ability to really work out what's going on dynamically and how to control things. It also made me think about the possibility that engineered cells could be used not only to treat cancers, but to prevent cancers in scenarios such as, you know, people who have germline mutations that make them at very high risk. You know, if we understand how we think some of these non-mutational transitions might be involved, say, in, for example, BRCA mutant individuals, right? We might be able to manipulate the immune system to simply reduce the probability that those transitions occur. I think that's an interesting thing to think about.
Yeah, it's fascinating. And as someone who builds perturbative technologies specifically for signaling with optogenetics, I think the possibilities on the molecular level are actually limitless, and that's just signaling. And then, you know, we saw what Mo can do with the both optogenetics but also epigenetic regulation. So that is very exciting. One question from the Miro board: how can, maybe more directed towards Arthur, how can spatial technologies help constrain or inform the types of mechanisms, for example, diffusion or immunity, that could control underlying growth control?
Yeah, that's a really interesting question, because that is one of the areas where there's clearly some things we don't understand yet, right? We know that tissues and cells can coordinate over different length scales in space, and we only partially understand certain length scales. So diffusion operates on fairly short length scales, but mechanical constraints can operate on somewhat longer length scales. Bioelectric constraints can operate on much longer length scales. Those are areas that are kind of very understudied. And it would be really nice to be able to sort of have a well-developed theory of cell communication over different length scales that would then help us sort of predict what we should be looking for, when we see, for example, the transition between a precancer and a cancer occurring at roughly a certain size. Why does that happen, and what does that tell us about what are the molecular mechanisms we might want to look into?
On that note, Arthur, can I just ask, what are the challenges with sort of bridging scales like that from a computational or an algorithmic, perspective?
Um, I, I don't think they're that bad computationally. I mean, accounting for mechanics is a little more complicated than sort of looking at diffusion, but there are plenty of good people doing it. I think the real problem is experimentally doing the measurements that you need to do to look at mechanical properties within tissues and how they evolve dynamically. That's much harder than looking at expression of molecules. So, the people in the developmental biology field are certainly putting a lot of effort into that. People looking at wound healing, regeneration, and so on. So it'll be interesting to see, how the cancer field takes that up as well.
Yeah, and I would love to see more interaction between the sort of more canonical, if you will, mechanobiology types and the more molecular cancer types, right?
Absolutely.
To your point, really. Yeah.
Okay, I apologize for the abbreviated discussion, but we will continue this in the breakout room. But I do want to keep us on time for the other speakers. So thank you both. That was really a fantastic set of talks and discussion, and look forward to continuing it.
Thanks.
Thanks. Bye-bye.
All right, I think we should just directly start the next one. Can you guys hear me well?
Yes, we can.
Okay, perfect. So I think that this session that just ended was a perfect beginning to what we're going to talk next, because Arthur was already talking about inter and intracellular conversations and how cell intrinsic and cell extrinsic aspects of tumorigenesis play a very important role. So we're going to move a little bit towards those arenas right now. So the very first speaker will be, let me find, so I hope I don't butcher the names, Cassandra Borziak. She's from the Memorial Sloan Kettering Cancer Center, and she's going to be talking about the environment plasticity axis in tumor initiation. Take it away.
Okay, I'm just sharing my slides. Are you able to see them? Okay, great. So hi everybody, my name is Cassandra. I'd like to start by thanking the organizers for the opportunity to speak here today about my work in Donna Pear's lab at Sloan Kettering on the environment and plasticity interplay in the earliest stages of tumorigenesis. So I think everything we've heard so far plays really nicely into my introduction here. Of course, this is the tumor initiation session. So the key question of interest is naturally, how do cells which are initially in a normal state end up in what we would consider malignancy? And I think Ken's talk really sets up what I was going to say here very nicely, which is that this is actually really a pretty ill-defined question, in the sense that we don't really have a very clear definition of what is normal, what is malignant, and exactly where is the line drawn between these things. Coming from the single cell field, where we think about things in terms of cell states and phenotypes, one perspective on this might be to say, okay, malignancy is just sort of a very unique cell state, and we can define it functionally based on the features that these cells have, like they might be more motile, they might upregulate certain programs to resist apoptosis, so on and so forth. And if we think of things in this way, then we can sort of reframe this question of how do cells transition here in the same light as we would consider for even a normal biological system like cellular differentiation. And so all of this is really to set up the notion that we can consider this a case of phenotypic plasticity. And in fact, this was added to the hallmarks of cancer in 2023. And is considered one of the core features of cancer cells today. So all of this is very abstract. We're thinking about definitions and nomenclature for these things, but of course, when it comes to a therapeutic perspective, ultimately what we need to do is define what are the sort of molecular events that are taking place that are driving this axis of plasticity. And as we've heard several times today, and if I were to ask everybody in this audience probably to name some of their sort of favorite molecular events that they're aware of that give rise to this, they're going to think of these genetic events, right? These driver mutations, which I think we would all agree are certainly key players in placing cells in a state that allows them to upregulate this plasticity. However, we also saw many examples already in this meeting that this is not the entire picture. We can have these mutations in entirely normal tissues and then never develop malignancy or have a long latency period before malignancy develops. And so what is it that we're missing? So from our perspective, we could, define multiple different factors here, but we focus in our lab especially on the epigenetic changes, which of course are going to be very linked to this idea of plasticity. This is what defines plasticity and lineage decisions even in a normal context, and changes in the environment such as inflammation, which may trigger these events, especially in certain genetic contexts. So for my talk today, I'm going to focus on the concrete example of pancreatic cancer initiation, which is actually very illustrative when it comes to the interplay of these three features. Pancreatic cancer almost universally begins with an activating mutation in this KRAS oncogene, and this mutation alone will place cells in what you might consider a precancerous state. However, it's typically not considered potent enough to actually immediately drive this transition to malignancy. Cells will be refractory to this transition for quite some time. And it turns out that if you have certain triggers, such as a case of pancreatitis, we see this in humans that do have some chronic pancreatitis are more heavily predisposed to develop pancreatic cancer. And we can also do this in mice. Actually, inducing pancreatitis will trigger the onset of initiation of the disease in this KRAS mutant context. And ultimately, these cells will be locked into a progression wherein they end up in these benign neoplasms and ultimately malignant tumors. And so where does epigenetics come in? Well, we showed recently in 2021 that this pancreatitis event really functions through a wide-scale reprogramming of the chromatin accessibility landscape in these benign lesions. Okay, and so sort of to summarize all of this, the main thesis of my work is that when we think about these cell state transitions in the earliest stages of tumorigenesis, we really need to consider the combined interplay of these three genetic, epigenetic, and environmental dimensions. And we also can analyze these and understand these in the lab by using genetically engineered model systems, as well as using single cell techniques, single cell transcriptomics, and epigenomics, which allow us to phenotypically track the progression of cells as they evolve through the transition and computational inference to get at some of these environmental features. So I'm going to give one concrete example, again, from this case of pancreatic cancer today. So in this case, we are using these genetically engineered model systems that have an activating mutation in the KRAS proto-oncogene, and we are taking a time series of samples from cells that just have this mutation after we actually induce pancreatitis in these mice within 24 hours and then multiple weeks later until we end up having these full-blown lesions. And then from the other side, we're using single cell technologies to be able to probe the axes of transition here at a fully unbiased manner. So here you can see what the cell state heterogeneity looks like in this T-SNE. This is including both epithelial and immune cells that we capture in the system. But the main thing that I would like you to take away from this is that if you plot the cells along their first fusion component, component, which is describing the dominant axis of variation in the data, the first thing that you're going to notice is that even within 24 hours post this injury event, you get this already massive shift toward this malignant state away from the normal pancreas. And this is all happening in concordance with the rapid shunting of normal pancreas programs and the rapid upregulation of pancreatic cancer associated programs. So all of this is happening extremely rapidly, well before tumors will have formed. And if we take a bit of a closer look at the phenotypic heterogeneity just within the cells that have a KRAS mutation, not in the normal pancreas, what we see is that we have really a vast amount of heterogeneity here occurring before we end up getting these tumors in purple. And if we go and look at what are these cell states based on some markers we can use for annotation, we were really surprised and amazed to see that each of these cell states has been described before, and in fact, has been validated as a cell which has potential to give rise to these full-blown tumors. And so all of this is to say that with the context of this KRAS mutation, very, very early on in disease progression, before you have a chance to accumulate additional mutations, before you've developed anything that you would even consider to be a pre-malignant lesion, you have all of these diverse phenotypes emerging, and we consider these to be high plasticity phenotypes that can give rise to malignancy. So how does this happen? How is it possible that we have this diversity emerging when we have similar genetics at this point in time? We used single cell epigenomics, in this case, single cell chromatin accessibility profiling, to try to understand what might be the underpinnings of this diversity. And we found a very interesting pattern. We noticed that for many cell states, their chromatin accessibility landscape is very, very well linked to their transcriptome, in that you have accessibility only in those regions which we see expression. But we also saw a number of cell states where you see this priming of chromatin near genes which are unexpressed, which we interpreted as a sign of increased plasticity of these cells because they can sort of rapidly change their expression programs. And in particular, these were often near genes related to cancer. We found that beyond them just being related to cancer, that many of these were also related to cell-cell communication. And so this was sort of the aha moment of this study, which was that we found cells that have this plasticity are also endowing themselves with greater potential to communicate with their environments through opening and general unleashing of these communication channels. So quickly, I'll mention how do we understand the functionality of this unleashing of communication programs. We can use information that we already have on which cytokines and receptors may interact with one another to build these complex communication networks between cell states using algorithms that basically attempt to address the statistical co-occurrence of these things in the tissue. And with this, we can do things like tracing paths of communication through the tissue. And most interestingly to us, we looked for feedback loops, which might be responsible for sustaining these changes which occur in the face of inflammation in this system. We found one in particular, which was driven between one of our plastic populations and Tregs in the environment. And we can even visualize this in space to see that these cell states are co-located spatially. And so to kind of summarize everything I discussed here today, we are considering the transition of tumorigenesis as a sort of multidimensional feature of these three factors. In this study, what we did was we at least probed one angle of this genetic axis by looking at KRAS mutation versus KRAS wild type. And what we found was that there's this sort of correlation or positive correspondence between the inflammation in the environment and the plasticity, and that plasticity enables you to communicate through channels which were unavailable before. In the future, we would like to expand these axes by looking at additional mutations, which is something that can certainly be enabled by different systems that we've already discussed today, such as organoids. I bring this example specifically because my co-presenter in the next talk, I do believe that there's opportunities for us to use the kinds of tools that Adam will talk about in the context of these organoids. And then finally, we want to look at these later stages of progression, which is also relevant, especially in the spatial context where we think about things like tissue integrity and how these can be affecting things such as metastasis and lineage transformations later on. And so with that, I would like to thank my lab, especially Dorena, who was my co-collaborator on the work I presented today. And I'm looking forward to discussing more at the end. Thank you.
Thank you so much. That was beautiful. There's a lot of questions already, but with no further ado, I'm going to introduce Adam Stevens from the University of Pennsylvania, just downtown from where I'm standing right now. And he's going to be talking about programming cell adhesions to interrogate the tissue organization and tumor development. Go ahead, Adam.
Okay, do you hear me? Hello? Okay, yeah. So I'm gonna assume you hear me.
Yep, we can hear you.
All right, do you see my slides? Yeah, okay, great. I'll start. So I'm Adam Stevens. I recently just started my lab at the University of Pennsylvania School of Medicine in the Department of Cancer Biology. Most of what I'm going to talk about today, though, was actually carried out in Wendell Lim's lab at UCSF and at the Cell Design Institute. And today I'm going to describe a receptor toolkit that we developed to direct cell adhesion. The long-term goal of this work is to better understand multicellular systems by tailoring specific cell interactions that occur within them. We're also interested in applying these tools to engineer synthetic tissues and cell therapies to study and treat cancer. So the biological process that directs how cells interact with each other is called cell adhesion. And it's clear that adhesion is fundamental in multicellular organisms just from considering its role in the formation and maintenance of tissue architecture. It also contributes to the generation of complex communication networks, such as the wiring of the nervous system. And furthermore, adhesion controls dynamic processes, such as how immune cells traffic to sites of disease within the body. And this movie is showing neutrophils passing cells adhere to the endothelial wall and then migrate into the neighboring tissue. An analogous process to this can also occur during tumor metastasis. And so during this, cell adhesion can contribute to tumor cells escaping the primary tumor site, migrating and entering circulation, and then localizing to sites of metastasis. The goal of this project is to obtain synthetic control over cell adhesion. We want the ability at the molecular level to program individual cell interactions, and then this should enable tailoring of complex multicellular tissue structures. In addition, we'd also like to characterize and control how cells migrate to tissue. We want to understand how cell adhesion operates at the molecular level in these processes, and also apply this as a tool to both model tumor organization and potentially intervene with cell therapies. So if our goal is to program cell adhesion, the most direct way to do this would be to express a programmable binding domain, such as a nanobody or scFv, on the surface of a cell. This would then direct that cell to bind a second cell if it's expressing the corresponding ligand. However, endogenously, cell adhesion molecules are a lot more complex than this. So along with having complex modes of extracellular recognition, what many cell adhesion molecules have in common is that upon binding, they undergo a signal transduction process whereby the intracellular domain will engage and or reorganize the cell cytoskeleton. And so for some of these molecules, this signaling domain will directly anchor to the cytoskeleton, which will strengthen the interaction between cells. Other adhesion molecules can locally decrease tension at a contacting interface. The result is a more expanded cell contact, which also contributes to adhesion strength. So what we'd like to have in a synthetic adhesion molecule is programmable extracellular binding, but then also coupled to having the ability to retain the signaling effects of the intracellular domain of a cell adhesion molecule. And to accomplish this, we used a chimeric engineering approach where we replaced the endogenous extracellular domain of an adhesion molecule with a programmable binding domain, but we retained the native transmembrane and signaling domains. This provides two levers to control cell adhesion. So the first is that the extracellular domain determines the manner in which cells interact. So binding can be homotypic or heterotypic. Specific ligand targets can be programmed depending on the choice of the scFv. And then also properties of binding, such as affinity, can be tuned. The second lever is that the choice of intracellular domain determines the signaling output. So there are hundreds of different cell adhesion molecules found within mammals, and many will lead to distinct signaling effects and distinct mechanical effects in the cell. And then I just also want to point out that this approach allows us to isolate and study how specific molecular properties within these receptors can contribute to cell adhesion. So to characterize the function of these synthetic receptors, we first expressed pairs in L929 mouse fibroblast cells. This is a cell line that's commonly used to study adhesion due to lacking expression of strong homotypic adhesion molecules. And within these pairs, we maintained a constant extracellular GFP anti-GFP interaction, but then varied the identity of the signaling domain. And importantly, we compared the synthetic receptors to a control that maintained the same extracellular binding interaction, but lacked any signaling domain altogether. So it has a tethering interaction still maintained on the outside of the cell. We made sure to match expression of these receptors, and this was to ensure that differences in adhesion that we were measuring were due to the receptors themselves and not differences in expression. And then we characterized cell interfaces using confocal microscopy. And specifically, one of the properties that we measured is the cell contact angle. And that corresponds to changes in interfacial tension that occur upon adhesion. And also this correlates with adhesion strength as well. So we observed native-like expanded interfaces with synthetic receptors that had signaling domains from molecules such as cadherin, ICAM, and integrins. And this is despite these synthetic receptors lacking the complex native extracellular domain. However, in comparison, the tether control interaction, it really only had a small point of contact. It didn't lead to the same expanded interface that we saw with the synthetic, with the signaling domains from native adhesion molecules. And so we conclude that the intracellular domain is driving the expanded cell-cell contact. We then characterize the relative effect formation. And to do this, we used a series of nanobodies that span 4 orders of magnitude of binding affinity. Once again, binding GFP. And so we see that with decreasing affinity, there's a corresponding decrease in contact angle and adhesion strength. However, even the low affinity binders with the synthetic receptors exhibited greater contact angles than 2 high affinity tether control interactions that lacked any signaling domain. And so we concluded that while the extracellular binding affinity is able to tune adhesion contact strength, the intracellular domain is having a dominant effect for adhesion. And so next we asked whether synthetic adhesion molecules were affecting the cell cytoskeleton. And so this could potentially explain these observed differences in contacts that I showed you before. We allowed L929 fibroblasts to spread on a surface coated with GFP, and then we fixed the cells and stained them with phalloidin to observe the actin cytoskeleton. With a simple tether interaction, we saw minimal cell spreading. That's in comparison to cells expressing receptors that had previously shown the expanded contact. Here, they're expanding greatly on the GFP surface, with actin spreading to the periphery of this expansion. And then some other signaling domains actually had a very interesting phenotype where they had a smaller central actin region of actin, but then also these individual protrusions that were pushing out on the membrane. And so what we saw is that these different intracellular domain classes where we're forming we're directing different cytoskeletal structures. And so this is being determined once again by the signaling domain. So just to slightly shift gears, because we can program specific cellular interaction partners using these receptors, we wanted to then take this and see whether we could build custom multicellular assemblies. This is really a first step towards building synthetic tissues. Using homotypic leucine zippers, we showed that we could program fibroblast cells to differentially sort into defined and distinct regions as determined by the receptors that are expressed in them. We can use combinations of orthogonal heterotypic interactions to specify patterning within a specific region. And then also we can design these molecules to directly compete with adhesion of endogenous adhesion molecules. So here we programmed a molecule to bind peak adherent, and we can see that cells expressing the synthetic receptor intercalate within the assembly, while those that do not are to become differentially sorted from the wild type peak adherent expressing cells. And so we can really build an array of different interactions. We can build homotypic, heterotypic, and also targeted binding within these groups of receptors. We then asked what happens when we take two distinct groups of cells, such as an MDCK monolayer and L929 cells expressing a strong homotypic molecule. So these groups would normally sort from each other, and then introduce strong heterotypic interactions of increasing strength between them. So how would this affect the resulting system? So we saw that with no adhesion or a minimal architecture. But when we started to introduce a stronger interaction through a coherent or an ICAM signaling domain, now the spheroids, instead of forming spheres, they flattened on the surface. And interestingly, with this ICAM signaling domain, we saw that a type of cooperative rearrangement where both tissues formed a lattice-like structure. And then lastly, I want to highlight a recent publication from a postdoc in the LIM lab named Toshi Yamada. And he took this type of tissue engineering approach to another level, where he used synthetic adhesion molecules to pattern the assembly of morphogen secreting organizer cells. And he used these secretor cells to differentiate mouse stem cells. And so he saw that by spatially arranging the organizer cells around the mouse stem cells, he was able to have defined morphogen production, which yielded complex tissue morphologies, including this type of beating. And so, with those last two experiments, I wanted to get towards what this tool might be able to accomplish in terms of tissue engineering. And just to summarize the talk overall, we showed that we can design synthetic adhesion molecules that program cell interactions. In turn, this can be used to organize multicellular structures. Some of the problems that I'm really interested in exploring next with these tools would be to potentially study the role of adhesion in tumor development and migration, characterize how changes in adhesion contribute to immune-excluded tumors and potentially engineer immune cells that can reestablish entry, and then also potentially try to improve approaches for cell therapy by increasing adhesion strength between T cells and target cells. And I'm out of time, but I have to acknowledge Wendell and Dan Fletcher, who are two of the main group leaders behind the work, and then also everyone else who contributed. All right. Thank you.
Thank you so much. So we have some time. I need my brain to calm down a little bit because this is so exciting. So I can see the potential, and I can see why both of you were set up to be in the same section. I would like to ask a very biased question to Cassandra first. So you were talking about changes in the chromatin axis because of the microenvironment. So theoretically, once there are these epigenetic changes, then it would not matter if the cell will be in a different environment, and it will be intrinsically driven to develop into a tumor. And so that's really just a thought, and I wonder, what do you think about it? I would like to just highlight some experiments done by Beatrice Means in the 70s, where she was showing terrible teratoma cells that were fully already tumorigenic, implanted into developing embryos, can generate pops that are mosaic and free of cancer. While if you retake those cells that were part of these pops and put them in the right environment, you will get cancer again. So if it were mutations and epigenetic cancer cell intrinsic, these microenvironmental changes would have not the impact that they do. So I wonder how you see those types of experiments with your results and your research.
Sure. I think overall, when it comes to this interplay, the first thing I'll say is that I do think there's a chicken and the egg problem. I don't think it's causal in one direction. I certainly don't think that it's sort of a inflammation happens, this opens chromatin, and then this is the end of the story. What we're seeing specifically is that inflammation happens, it remodels the landscape, but a big component of that remodeling is opening these channels of communication. I think at the stage that we were looking at in the work I described today, there's still a very heavy dependency on inputs from the environment. There's some evidence from follow-up studies that sort of this dependency maybe doesn't last forever. So I do think that in this case, these cells are p53 proficient. They are still having lots of checks on their growth and their division. I do think that there's some evidence now that later on, once you lose p53, maybe then in this situation, your environmental dependency is much lower. So I sort of think that what's happening a bit speculatively is that at this very, very early stage, the tumors are dependent on this input from their environment, or they're not even tumors at this point. The tissue is dependent on this input from their environment to sort of phenocopy something that they will eventually become more independent later on when they have for sort of full-blown malignancy. But yeah, I think the reality is that it's a very complex interplay. It's not just a unidirectional shift. And I definitely would not argue that at this stage you could take those cells out and then they will be fully proficient, able to grow into full-blown tumors.
So there's more stuff we can do together.
Yes.
For Adam, so the experiments that you're doing remind me a lot of the classic chimeras to start studying clustering of receptors. Probably those were done in the 80s and the 90s. And so the signal transduction within the intracellular domains are intact, and people were using chimeras to use ligands of different receptors to cause clustering. And you're using a very similar idea, but to do cell-cell interaction as opposed to cluster. I wonder whether to fully understand the development of the tissue in these multicellular environments, you're missing the clustering of the receptors, because I understand that you can do the different type of adhesions based on what you're choosing between the two cells. But what about in the intracellular clustering and complexes? Any thoughts as for how that can be accomplished?
Yeah, so I think that's clustering is really one of the main mechanisms where that leads to signal transduction from the outside of the cell to the inside of the cell. For some of these receptors, even without the extracellular domain, we see significant clustering. So these are adhesion molecules that are known to recruit scaffold proteins. We see them cluster. We're essentially they form like because we have GFP as the extracellular domain, we can see how the receptors are localizing on the membrane. And so some of them that are known to recruit scaffold proteins form almost a focus of GFP, where it looks like they're clustering to a point, while others don't do that. They're more spread out on the membrane. There's also the point domain is contributing to clustering. And so for cadherins, that's the case, where there's Barry Honig's shown that these can cluster in cis through interactions of the extracellular domain. And that's true for a number of other adhesion molecules. And I can be introduced within the synthetic system, where we could potentially build extracellular domains, parts of the extracellular domain that have clustering components. And then we can not only, have clustering, but actually probe what's the difference when you have a receptor that clusters versus doesn't cluster, and one that has adhesion versus not adhesion, because another contribution is actually tension that's applied to these receptors. So there are multiple going on here, and I think actually looking at clustering would be really interesting to do.
Yeah, so what you're saying reminds me also of Alpha Yap's games with cadherins, coating, and combining with Christian's putting the little different clusters of the whatever receptor would be used. And
Yeah.
so I think that you're taking all of this to the next level, and it's very exciting. There were a couple of questions set up over there in the Miro, I think it's called. And one of them, let me find it right now. Oh, yes. So there was a question about how to distinguish in the epigenomic changes, which of those are causal versus consequential. So it's probably for Cassandra.
Yes. Yeah. So, I mean, the way that we framed it in this paper, which is maybe a slight simplification, of course, we have to make these in this type of analysis, is that you think of the epigenetics as sort of the programming of the cell, right? So we're dealing with the two measurements here, the epigenetics and the transcriptomics. So our notion of phenotype here is coming from the transcriptomic data. This is sort of what we're using to define where on this axis of malignancy we're falling. And I guess the premise there is that this is somehow closer to the functional cell state than the epigenetics, in that this is the genes I'm expressing, this is what my function is. So I would say that the way we've envisioned this is that the epigenetics is seen as the programming. So there's an assumption that there's this causality between this and the transcriptomics, and not necessarily the other way around. However, of course, same answer as to the last question. This is a bit of a chicken and the egg, and there's obviously feedback between these things. But in terms of defining plasticity, the way that we view it is that the epigenetics is the programming, the potential that you have to access these different transcriptional programs, and therefore different phenotypes.
So I know that there is a lot of questions we can then take for later that are a little more specific, and Sandra was showing me that she may have for later. But at this point, I would like to know from each of you, how do you see working with each other? Because you're not obviously, but yes, potentially very much complementary of each other.
Yeah, I mean, I can start by saying that when I went over Adam's work and heard his talk, I'm like, this is the holy grail of some of the stuff that we do, because we are limited to this kind of, going back to what Ken said, this very measurement-based work when it comes to tissue architecture, and this would be something that would give the potential to do more intervention-based work. I think the question that I have is, thinking about this, especially in the context of cancer, one thing that comes up in my work over and over again is that cancer is sort of this like controlled chaos state. In our case, what we see is that, it's not necessarily a specific adhesion interaction that gets switched on that's relevant, but rather these cells are now just chaotically expressing tons more adhesion molecules and cytokines and things that are not typically present in the normal state. So I guess the question that I have is kind of, how complex can we go with the synthetic system? How complex should we go? And to make it relevant for the stuff that we're studying with cancer initiation.
Yeah, that's a really good question. I think having the ability to perturb the system in a defined way is where I think there could be a lot of overlay in the types of work we do, where you could use a synthetic system like this to introduce one, or maybe if it's more complex, 10 different adhesion interactions or some other type of perturbation like we saw in the previous session, and then come in with the types, the multi-omics or complex chromatin probing readouts, and then ask, what's the effect of a defined perturbation, like synthetic perturbation, on the cell state. One of the problems that I was really thinking about with your work is tumor metastasis, and whether we can identify the state of the cell that leads to metastasis to a distal region, and because it could be, it might be somewhere along that landscape that you're observing, right? And the question is where, and does that also define where the cell is going to metastasize to? What tissue might be hospitable for it? I think that's a question that I'm really interested in. I think it potentially has like overlay in both areas.
Absolutely. I mean, there's a lot of work also at my institution that I think everyone's favorite gene in the metastasis realm at my institution is L1-CAM, which is right up your alley. And yeah, I think that there's metastasis is an especially relevant example when it comes to loss of tissue integrity. How does that impact the transcriptional state? And then, of course, the tissue organization is a very key player in that.
So maybe one last question for both of you. So based on recent really exciting work from rapid autopsies from individuals that die from nothing related to cancer, Marina Pascali-Mangliano showed very nicely that the majority of us, if not all of us, are walking around potentially with precancerous lesions in our pancreata. Yet we know that pancreatic cancer is a very, very rare disease, and the majority of us will not develop this cancer. And so it's obvious that there is something intrinsic to the human that is not part of the precancerous lesion mutations per se, and perhaps is the epigenetics, but we're not. I don't think that has been looked into. So how can you use your approaches and your type of experimental thinking to try to address that question? Because potentially, and pancreas is just as an example of a model, because potentially based on both of your expertise, we could, and what we heard so far today, we could really intercept and prevent all of this from ever unleashing. And so I wonder what are your thoughts for that?
Yeah, I can start. I mean, I think that this is exactly, partially the question that we tried to address with this, although it's in mice. So, we have this limitation naturally, which we've already discussed today. I do think that we can't model every single aspect of the human disease, of course, in mice. I would say that I'm of the belief that this environmental situation is really what is the key difference, and why some people, and pancreatitis is one very potent example of that, but I'm sure that there's many other causal factors. And yeah, I think, moving toward, there's people trying to develop these pancreatic organoid systems. I do think that is one area of potential experimental advancement in this realm. And yeah, that's hopefully we can figure it out.
And yeah, and just kind of along those lines, something I think about is defining organization of different cell types and asking how that might affect the cells within that group of cells. And depending on the arrangement, you might see different metastatic potential or just invasive potential. And when it comes to intervention, you might then introduce some type of therapy or cell therapy that's different depending on the state that you're starting, that you observe initially. So something along those lines are problems that I think about as well.
All right, I think there is a lot of really interesting questions that we can bring up for later. And so I'm going to try to summarize and see how we can then discuss that later on. But thank you so much. This was really, really exciting.
And thanks to
Thank
organizers.
you.
Thank you all very much. We're going to move into a break right now. Please be back promptly at 3 p.m. where we'll move into our next session. And we're going to post a slide up that shows the break time. But if you want to draw your attention to the left side of the slide, please also take a look at an upcoming opportunity that may pique your interest. That's really an extension of the activities that we're talking about today. So breaking until 3 p.m. and we'll see you all soon. Thank you so much.
All right, I'd like to welcome everybody back. This is Laura Heiser. I'd like to also introduce the moderator for our next session, Dr. Bianca Dumitrescu, coming to us from Columbia University. Bianca, as you're ready, please, excellent, unmute your video and audio. Dr. Dumitrescu has been at Columbia since 2023 and is affiliated with the Institute for Cancer Dynamics. So very timely set of expertise that she brings to us in moderating this session. And with that, I will hand over to you. Thank you.
Hi, thank you so much for the introduction. It's my pleasure to drive the conversation for this third conversation tonight. Tonight, I'm in Zurich right now, so it's night for me. I guess it's during the day for you all. This section is going to be about systems and synthetic biology, and we're going to focus on underlying principles that connect both in vitro and in vivo models, and we're going to look at a couple of questions ranging from how to perturb systems and what to observe from them. The first speaker in this section is Caroline Schutt-Ibsen from Oregon Health and Science University, and she's going to be talking to us about 3D programmable engineered tumor models enabled by ultrasound responsive biomaterials. I'm very excited for your talk, Caroline.
Great. Can everyone hear me and see my screen okay? All right, wonderful. So first off, I want to say thank you very much for the opportunity to present in this workshop today. It's always really exciting to hear from these different cross-disciplinary perspectives. So my background is in tissue engineering and biomaterial science, which really informs our approaches, building tools to advance 3D biofabricated in vitro tumor model systems that, in our case, are enabled by responsive biomaterials. So hopefully today I'm looking to stimulate some thoughts on how these 3D programmable engineered tumor models can be leveraged to address some of the important biological questions we've heard about today that are generated by systems biology. So 3D biofabrication methods like bioprinting, for example, allow for building ever more spatially complex features into engineered tissues that can better mimic the tissue architecture. So these approaches have been utilized extensively in the field of organ tissue engineering and include printing 3D anatomical structures with intricate features like these cardiac models. And these approaches are also now being applied to mimic the biophysical parameters of the tumor microenvironment, including organ-on-chip type models, which we've heard about, that include vessels and tumor organoids and can allow flow. And also bioprinted multicellular structures that can control the spatial arrangements of multiple cell types. And these structures are built up of multiple cell-lated filaments, which are part of a 3D scaffold matrix. And these techniques are really important because, as we've been hearing about all day today, the spatial geometry and also proximity to different stromal components can have a really significant influence on cell behavior and also progression to malignant disease. But along with this spatial complexity, there's also a critical need to coordinate temporal complexity as well. And advancements in these 3D in vitro model systems are going to require the ability to direct cell processes in time within these structures with spatial and temporal control after they are formed and also throughout their maturation. This would enable the formation of healthy tissue structures containing functional cell-cell junctions and cell matrix interactions, and also intracellular communication prior to the stimulation of tumorigenic events. And a key tool for achieving this type of in situ genetic manipulation of cells is to do in situ genetic manipulation of cells to affect their protein presentation. And this is really a cornerstone of biology, but it's primarily been designed for 2D and not 3D culture environments. 3D cultures often present some sort of barrier for traditional transfection agents like viral particles, where the scaffold matrix that builds up these models hinders diffusion of these traditional vectors. So this has kind of motivated the core theme of our group to engineer 3D in vitro tumor models that can recapitulate spatial features of the tumor and can also be genetically manipulated over time, creating, in a sense, a 4D tumor model. So to achieve this, we developed 3D cell culture materials that are responsive to externally applied stimuli. And we integrate these materials with biofabrication techniques such as 3D bioprinting and also organoid and spheroid cultures. And in particular, we use focused ultrasound as a unique external stimulus, which has some really unique properties for remote manipulation. So some of the core areas that our group works in includes developing platforms for 3D cell culture and bioprinting with responsive hydrogels, where we aim to recapitulate and manipulate cells in 3D tumor microenvironments. And we do this through the design of nano and micromaterials, which we leverage for environmental sensing and also triggered gene delivery in our models, as well as the development of ultrasound stimulation platforms. So as I mentioned, focused ultrasound has some desirable properties as a remote stimulus for gene delivery to cells in 3D culture. It has significant tissue penetration depth, up to around 12 centimeters at the frequencies we use, and also really tight spatial localization, and is also safe to cells at the low intensities that we're using. And it can be applied non-invasively and focused to a tight spot within the tissue. In our core technology, we've developed ultrasound responsive cell culture scaffolds, which we call sonoscaffolds, for non-invasive localized gene delivery within 3D tumor models. So this approach can localize transfection of oncogenes within 3D tissues. So in our system, we load these ultrasound responsive microbubble gene delivery vehicles into a hydrogel matrix along with cells, and we can then use our focused ultrasound beam to activate the microbubbles and control the genetic manipulation of a small subset of cells within the matrix at a desired time and 3D location where only the cells within that ultrasound focal zone will be transfected. And here you can see on the right, one of our collagen-based hydrogel matrices loaded with these green gene delivery microbubbles within. So using this platform of transgene-loaded microbubbles, when we expose a region of the 3D tissue construct to focused ultrasound, this creates a localized region of transfected cells in the ultrasound focal zone, expressing our gene of interest. We can program different patterns of ultrasound exposure into the 3D model using our automated robotic systems, where here the ultrasound beam is being focused up into this hydrogel. And this allows us to create user-defined patterns of transgene expressing cells within the matrix. So we've extended this technique to show ultrasound localized transfection of DNA coding for VEGF growth factor. So here on the left, you can see the transfected cells in the center ultrasound activated region of the tissue construct, which is also evident in this 3D view of the side of the hydrogel, where these green dots are now cells, transfected cells secreting this VEGF growth factor. So in the context of breast cancer, we're leveraging this system to transfect HER2 proto-oncogene, which we pattern into breast epithelial cell containing constructs. So shown here in the circle are the transfected cells in the approximately millimeter region of ultrasound exposure. And if we look within this region, we can see breast epithelial cells, these are MCF10As, now expressing the HER2 protein on their cell surface in green. We want to use these models to understand how do these HER2 overexpressing cells influence neighboring cells within the 3D tissue context, and how can we intercept these interactions in early stage disease. We're also interested in looking at differences between wild type HER2 and its different isoforms, which perhaps we will hear a bit more from our next speaker. We also are using this system to manipulate breast epithelial spheroid cultures. So here we can transfect a subset of these cells to overexpress HER2, and then evaluate how these first few cells influence the cells around them under different matrix microenvironment conditions. So using our technology, we've now shown that we can transfect individual cells within breast epithelial spheroids, where here we've transfected just a few cells per spheroid, and then shown the ability to modulate the number of affected cells with the ultrasound stimulus applied. So one of the intriguing things we find when transfecting these spheroids with HER2 is that often these HER2 positive cells will send out protrusions into the spheroid that start weaving their way around the healthy cells in a way that's also been observed in focused ion beam electron microscopy images of HER2 positive patient tissues. So this is a behavior that we're now starting to study more carefully in these model systems. We can also extend these ultrasound activation techniques to 3D bioprinting, which allows for creating more complex 3D architectures. So here we integrate our gene delivery vehicles into printable bioinks, and we can print cells into 3D structures to create tubular or vessel-like structures that you see here. And these printed bifurcated constructs contain cells, and when we apply ultrasound, we can deliver genes to a specific spatial region of our choosing, altering cells at specific locations within a heterogeneous tissue structure. We can also utilize bioprinting's ability to layer different inks to activate multiple gene cargoes in the same print. So in this print, filaments containing microbubbles for M-Cherry expression are alternated with filaments containing GFP microbubbles. And by rastering the focused ultrasound in a line across the construct, we selectively activate multiple inks, and we can deliver distinct genes to our bioprinted cells in a region of our choosing. So in addition to activating different genes in distinct regions, we're also building a platform to multiplex or temporally control different gene delivery events in the same tissue region, which can be used to look at how the order of different mutations could affect outcome in a particular microenvironmental context. We're also building models for studying interactions with the endothelium during tumor progression, where here we create microfluidic chips with endothelialized and perfusible hollow channels. And using a different type of bioprinting, robotic aspiration, we can deposit breast epithelial spheroids at controlled distances from the vessel, with here showing 3 spheroids placed adjacent to the vessel at increasing distances. And we can endothelialize this system to give us spatial control over interactions between a mammary epithelial spheroid and the vasculature. And then a final capability that I'll just mention today of these biofabricated systems is also the ability to embed real-time sensing elements into these structures. As oxygen tension can play a key role in cancer progression, we're integrating microenvironmental oxygen sensors into these models that allow us to map oxygen tension in the spheroid and at depth within a 3D matrix, and also report longitudinally on oxygen tension over time. So using these sensors embedded in 3D models with triple-negative breast cancer cells, on the right, you can actually see the hypoxic core initially present within the spheroid. And in the middle column, we see that these cells remain glycolytic over time. So this tool provides a way to monitor oxygen tension and follow cell processes in an intact 3D model. So to summarize, a couple of the tools that we've developed, our ultrasound-mediated technique enables spatial and temporal control of gene delivery in 3D biofabricated model systems with application for controlling oncogene delivery in engineered tumor microenvironments. And with these systems, we hope to have a new tool to study coordinated cell processes within 3D tissues. So ongoing areas of interest include integrating immune cells into these models, and also looking at manipulating patient-derived organoids, and looking to ask questions about how does the matrix microenvironment properties in stromal cell neighborhoods affect competition between different clonal populations in the early stage. So with that, I of course want to acknowledge our team and funding sources, and looking forward to discussing this further.
Thank you, Caroline. Now, our next speaker might be Josh Snyder. Josh, are you there?
Yes, I'm here.
Oh, wonderful. So you made it in time. Cool.
Yes, I just made
it. This is fantastic. It's great news. I was prepared with a lot of questions for Carolyn for us to have a coffee chat. But Josh, it's great to have you here. So our next speaker is Joshua Snyder from Duke University. He's going to be talking about illuminating false rules of cancer progression in vivo, to progress or not to progress. That is the question. A very deep question indeed, and I'm so excited to hear more about it.
Yeah, you may have to ask her a question indeed, because I have an update that won't let me share. So I have to log off and log right back on. So I'll be back in 30 seconds.
Sounds great.
You can display.
Caroline, if you're around, actually, until Josh comes back, I'm gonna warm us up with some of Fold's rules. So I don't know if you can hear me, but one of the questions, one of the rules is that progression follows gradual steps or continuous or discontinuous steps in progression. So in your system, are you able to apply discontinuous kind of ultrasound stimuli? Do you also have a way to do it continuously?
Yes, so that's an excellent question. And one of the things that we're looking at modeling in our system is trying to really explore what we can do with the temporal component. So thus far, we've looked at, can we activate a single mutation, or can we deliver a single genetic payload within our system? But really, it's going to be interesting to look at the behavior of different clonal populations and what happens. For example, can we ask questions about Does the order of mutations matter, or does the order of activation of these clones matter? And how do the properties of the microenvironment, either in terms of its stiffness or looking at different microenvironmental proteins in the matrix, how do those affect clonal competition? So building in the temporal element to these more spatially complex in vitro models is really something that we're interested in pushing.
I see. That's really cool. I'm particularly, I see Josh is back. So I guess we'll continue this. Josh, we're just updating of what Folds laws rules are. So we were just doing a little bit of a summary before your talk. So I think you're muted. So thanks, Caroline, for the impromptu session.
So my computer's not allowing me to share. I sent my slides. Hopefully someone can bring them up.
Yep, we'll get them popped up right now. Thanks for your patience.
Good, thank you. So I'm super excited to just update you on some of the work that my lab has been doing. And I think really what my work has turned into is kind of a little bit of a history lesson in where tumor progression has come from, if you will, and where we think it's headed in the future. So I hope to convince you of the reason for understanding this really important term and how we're really trying to tackle it in the modern molecular era. So next slide, please. And just click through it, open them up, all the cartoons for me. Thank you. So, this is really a really important problem. It's been since, the early 50s and 60s that a really important set of literature was initiated by a surgical oncologist by the name of Ian MacDonald. And what he postulated was that the preclinical phase of the tumor's growth and development occurred over the span of, approximately 8 years. And so more or less, there's this kind of missing period of tumorigenesis where he quips that the biologic balance between the neoplasm and the host has really already become established long before the clinician has any influence on the process. And he based some of this on some very interesting epidemiological evidence where there was a public health campaign to encourage women to self-exam for the evidence of lumps in their breast and then go to the clinic as quickly as possible. And what he found was that kind of early screening paradigm, you can see that there was an increased incidence of breast cancer and early stage breast cancer, which was the goal, but there was really no remarkable decrease in mortality. So again, he argued that from this, you could say that, a lot of the progression, a lot of the interesting aspects of the disease have been initiated very early. We can, of course, actually see a very similar problem today, fast forward to 2016, and you can see the increased detection of screen detectable mammographically detected breast cancers has increased, but the levels of metastatic breast cancer have not really decreased as dramatically. And so it really argues that there's something really important that we're missing very early on. And in fact, if you actually look at this really interesting study from this past fall, what it shows is that the majority of breast cancer specific deaths, because of the sheer volume of these patients, is really due to the early stage diagnosed breast cancers. So what is happening early is very important. Next slide. Next slide. Yep, there we go. And so Leslie Folds had actually remarked on this in the late 60s, and he says that the clinically recognizable lesions represent only a fraction of life history of the disease. And he goes on to say that there are certain stages of irreversible qualitative changes in one cell or multiple cells that results in an irreversible change in the characters of these cells, so their phenotypes. And this is what he calls progression. And we have to remind ourselves that progression is not merely the extension of the lesion in space and time, but it's really a dramatic change in the lesion itself. Next slide, please. And how can we think about these characters? Well, I think, if we kind of update Leslie Folds' terminologies, I think we can think of a character as really one of the well-described hallmarks. It could be anything from evading a growth suppressor to avoiding immune destruction, etc. And it's really these characters then, when defined, we're able to actually quantify them and really ask how these characters might change dynamically over time. Next slide. And what Leslie Folds gives us in this, reviewed here in this next slide, are 6 rules of tumor development. And at the top right, what I've shown you is the initiation phase. That is this little square where there's an oncogenic mutation or DNA damage, what have you, occurring in a cell. And then that cell has to somehow develop or progress into into a malignant disease. So he gives us 6 actually rules, which are really fun to think about. So rule number 1, if you can click through these with me, rule number 1 is that tumors independently progress. And so what that means is that in this kind of field of gray boxes here, we can't really tell which box is going to progress to the next stage or not. Number 2, we don't know if we think about these characters, if we define them like evasion or proliferation, these characters seem to progress independently of one another. So in other words, there is no rhyme or reason or sequential order in which they must occur. The third one has ramifications in screening, is that progression occurs independent of growth. So these malignant attributes, these characters can be acquired independent of size, which can really make clinical detection very challenging if you progress to malignancy before detection. Next, these characters can progress abruptly or gradually. Next, the characters can independently or follow alternative paths. They all reach the same stage, but they might get there in a different order or a different sequence. And then lastly, the progression of a character need not occur within a patient's lifetime. So some lesions may be benign forever, and it may not necessarily progress to the next state. Next slide. So what we've been trying to do in the lab is to really systematically work through these rules. And so we've built a suite of rainbow modeling technologies. We call them cancer rainbow mice, where we knock into the mouse genome multiple fluorescent proteins tied to multiple oncogenes. It's a simple Cree approach that activates these oncogenes, and then we can lineage trace and assess fitness of cell types and growth and progression in vivo. And we've published multiple papers on this topic on the right. Next slide. So this is just some images showing how we can look at oncogene activation in the mammary gland on the left, in the intestine in the upper right, and a new pancreas model, set of models that we're working on the bottom right. Next slide. So one of our favorite models is this HER2 model. We are expressing, if you can click for me to show the whole slide, we're expressing multiple isoforms of the oncogene HER2. The full-length wild-type isoform is shown there on the left. And on the right, we have an exon 16 splice isoform that lacks 16 amino acids in the juxtamembrane domain. And on the bottom right, we have a form of HER2 that lacks almost the entire N-terminus, called P95. So we've expressed these 3 isoforms in one of our models. Next slide. And what that enables us to do is to express 3 different human HER2 isoforms. Next slide, and then lineage trace the effects that these isoforms have on tumor development. And what we get is some really striking images like this here on this page. So mice that are 10 weeks old, you can see in the entire mammary ductal epithelium, these cells have recombined and a very large field has been initiated. So you see all these cells expressing an oncogene throughout the entire epithelium. But yet tumorigenesis is still an incredibly rare event. So only a fraction, a very small fraction of these cells will ever go on and progress to an actual tumor. In the middle panel, you can see a mouse 20 weeks of age. When we euthanize and image the entire mouse, you can see large tumors, and then down towards the bottom of the image, you see that little yellow dot. You can even see some very small tumors. Those small tumors are not palpable, and the large tumors are. But what you see is on the far right is that despite having such a dramatic advantage, the yellow isoform delta 16 is actually not all that much more tumorigenic than the P95 isoform. So the extent of the field does not really determine the extent of the tumorigenic process. Next slide. And so how can we measure this process then? How can we measure going from an oncogenic cell, that initiated cell, to a screen detectable lesion, to a symptomatic tumor that we can palpate and feel, all the way out to metastasis? And that's really what we've been doing over the last few years. Go ahead and click. And what you'll see is ways that we can do that. So we can look immediately after oncogene activation. Click, please. And then we can see using imaging, we can look at screen detectable tumors, which are those very small dots you see in the lower left. And symptomatic tumors, we can actually palpate and feel. And then lastly, we can image whole organs to look at the presence of disseminated tumor cells and metastatic lesions. Next slide. So we've started to really collect a wonderful set of data where we can capture across thousands of tumors, and we've done this and recently published on this, shape and histology of tumors. We have collected more and more single cell analyses of these tumors. We're of course doing a significant amount of spatial transcriptomics on these tumors. We're really starting to measure, I think, in a really deep way, all the characters of these tumors throughout the progression cascade. Next slide. And so in our most recent paper, what we've done is we've actually analyzed field growth in these tumors. So that is how quickly the cells are growing within the duct. We can analyze the number of screen detectable tumors shown here in that, that microscopy image beside the microscope. And we can also look at large palpable tumors. When we put all of this data together, we've built a set of ordinary differential equations, and we can start to ask questions, well, where is the defining rate limiting step in this process? And really what we can show is it's that growth from the field to this, what we're calling the occult period. This is the earliest nascent lesion that we can identify in the gland. This seems to be the rate limiting step. And it's actually in our hands in this P95 story. The P95 HER2 isoform is much more adept at crossing that first barrier. We're also able to look at cell state changes early, competition changes between the isoforms, dissemination potential, and metastasis. Next slide. And in the end, what we can show, if we can just click through all these very quickly, 6 clicks for me, we can really begin to look at the independent progression of tumors. That is, how likely is it that an unpalpable screen detectable tumor will progress to palpable ones? We can look at progression of characters like field growth. We can look at progression of, independent of growth. So this is looking at screen detection before or after progression to invasion. We can look at abrupt or gradual changes, alternative paths of development, and we can even simulate how many cells does it take to actually build a tumor at any age in the mouse's lifespan. Next slide. And so then that really raises some big questions. How can progression be intercepted? How can we build models that predict when or where these different progression steps are likely to occur? With the tall challenge that Leslie Folds leaves us with, that no tumors are alike, and generalizations of cancer inevitably break down. Next slide. So I'd like to thank all the people in my lab who contributed to this work. And then last slide, if you click one more time, we have a bioRxiv submission I was hoping would be available. But if you can click one more time for me. Where we're going is we've actually built a suite of fluorescent protein tools, and we're actually looking here at about 40 different fluorescent proteins where we're using UMAP to disentangle their hyperspectral signatures so that we can do not just 3 colors at a time, but maybe 12 or 15 different lineage tracing events simultaneously. Thank you.
Thank you both for fantastic talks. Your talks offer really a complementary view on how spatial context can shape tumor behavior, one through engineered microenvironments and the other one through spatial transcriptomic analysis in this beautiful rainbow kind of setting. My question is to both of you. How do you envision integrating your engineered models with spatially resolved molecular readouts to test hypotheses, the actual hypothesis that Josh actually formulated towards the end? Do you think they're complementary? How would you go about integrating them if you had time and funding and resources?
Sure, that's a great question. So we think a lot about opportunities to integrate these bioprinted or spatially designed in vitro models with spatial omics technologies to be able to further investigate the type of spatial interaction patterns between various cellular components in the tumor microenvironment. I think these technologies are really well poised for integration. And I think that's going to be a key opportunity. We spend a lot of time fabricating or building these purpose-built, spatially complex in vitro models. So in some ways, it's a shame to have to take them all apart in order to do single-cell analyses. So I think there's a really compelling use case for being able to use spatial transcriptomics technologies on these biofabricated in vitro model systems.
Yeah, and I love the idea of, as we get more and more spatial data from humans and our mouse tissue, our mouse experiments, using something like Caroline's built, where you could actually start to, what I was thinking about, Caroline, as you presented, was being able to recreate those spatial features that you see maybe in a static slice or a static specimen, if there's ways to actually recreate that three-dimensional and spatial geometry that we identify, then we can actually image it live and in real time. And I think, being able to do that, I think is going to be so important. And it's something that, the organoid technologies have been fantastic, but, again, that's disrupting the whole tissue and putting it back together again and hoping it recapitulates the geospatial constraints of the actual, of the tumor. And so I think that's going to be really interesting to see potentially how you could use something like Carolyn's technology to do that.
Yeah, it would be fascinating to investigate the findings that you found so far, Josh, with the differential progression between these clones and what's happening during that really critical stage pre-detection. I think that's an exciting opportunity for these in vitro models that can incorporate human cells and patient-derived cells and actually rebuild some of these interesting spatial or architectural features that might be important in influencing that progression during the early stage.
See. One follow-up question on this, kind of tying back to some of the previous sessions. Also, do you think your models can incorporate matrix composition or vasculature or mechanical cues? And do you feel those can have an important role in understanding whether some of the events you see, for example, Josh, are stochastic or deterministic or synchronized across different spatial domains? What's the role that mechanics plays in your setup?
Well,
Yeah, go for it, Josh. Yeah.
No, I think, I love to hear what Caroline thinks about it, but I think, one of the things that we're really intrigued by is this movement from like kind of being a cell in its normal physiological location to being in a slightly new environment, and is really important. And the mechanical or wounding like responses that occur during that step, I think, are instrumental. And where we think the progression actually occurs at its very earliest point is whether or not a cell decides to live inside of the fat or decides to live inside of the duct, right, is a really important early step that I think having some of these hypotheses from the in vivo, you can then go back to Carolyn's or similar technologies and start to ask those questions.
Yeah, just building off that, I think there's a really important interplay where we can take these findings from the in vivo model systems and then recreate aspects of that 3D spatial architecture to get at answering some of these questions about does location within the ducts or adjacent, does that matter? Does the particular neighborhood of stromal cells in the surrounding area matter? What about during if you have a stiffer or more fibrotic matrix structure, or if you have particular matrix proteins in there, like fibronectin has sometimes been implicated in having a role in these progressions. So I think one of the advantages or one of the things that we can contribute with these in vitro models is being able to swap out or systematically look at the influence of these different components and do so in a 3D architecture.
Thanks. And before we go on the break, there's one other question that I wanted to ask. So there was a really beautiful question a few sessions ago about length scale. So if we think about diffusion models, that's a length scale. If we think about how mechanics is read, that's another length scale. What are some length scales that you feel could be studied in your system? Can all of them be studied? Are there any particular biological or engineering considerations when thinking about that particular setting? Caroline, I guess this is for you.
Oh. Yeah, I think we tend to think about things in terms of cell neighborhoods and proximity to different geometries and architectures in the tissue. So at least with the techniques that we're building, speaking from my own perspective, we're thinking about kind of cell-cell interactions and also kind of tissue structure level interactions. But that's where we start from modeling in these 3D systems. I think there's also opportunities to go more into the single cell level and look at what's happening intracellularly that facilitates these interactions. So I think there's multiple length scales that are applicable, but I think we can build these spatially complex systems to interrogate cell neighborhoods and things more on that length scale.
Thank you both. I guess it's time for the organizers to encourage us to go to breakout discussions. So I let them take over. But thank you very much, Josh and Caroline, for a nice conversation.
Thank you, Bianca.
Yes, I'll jump in. Thank you so much for a nice conversation. So now we are going to wake you all up. We're going to test your ability to follow instructions and follow some details in WebEx. So we're going to post a slide here. And what is going to happen next is these 3 conversations that we've had throughout the day, you're going to be able to select which one you want to take a deeper dive into. So a little bit more engagement with the speakers, and then a chance to really expand upon the opportunities that have been identified and start to more deeply explore those. So technically, how to do this, these breakout rooms will open up. You will be able to click that participant window. You can see that person at the bottom there. If you click that, you should be able to see the breakout rooms. Josh, are they open? We'll just have to get them opened up for you, and then you'll see them at the top of your participant list. You'll be able to click and choose a session. And again, those will be aligned with the 3 conversations. So I will get those breakouts started. Josh, I can't start the breakouts while the slide is up. Perfect. Okay. Yep. Breakouts are, no. Carolyn, I'll be back.
So if everybody
no,
can just.
I am still in the main room. Okay. Sorry. Yep. So for those of you who are speakers or moderators or going to facilitate, you'll be automatically ported into those conversations. And then the rest of you, it's up to you to click that button to select which conversation you want to join. So pull it up, click on it, and then join for further discussion. So best of luck, and we're ready to see all your technical skills together. I'll see you in a room shortly. As we're coming back, I can just thank you all quickly for a more circuitous adventure than we had planned. So we really appreciate you working through these issues with us, and hope you at least had a little bit of a chance to chime in and engage with the group a bit. Thank you for fighting through that and helping to inform how this goes the next 2 days. We really appreciate it. And I think Laura and Zeb, when you're ready, I believe everyone's pulled back if you want to get things kicked off for the final panel.
Excellent. So we've invited a few people to join us in discussion. If you can turn on your video. And I think the AV people know who you are, so then you will be put in the spotlight.
Michelle, this is Sharmi. Do I need to be here?
Feel free to drop off, but we'd love to have you listen in too.
All right, thanks. I'll drop off. Thank you.
All right, well, while the others are joining, I don't see them yet, but I imagine they will pop up as they're brought forward. Our plan here for the next little while, just to close out the end of the day and reflect back on some of the ideas that we have discussed together as a group, and ideally pull in as well some of the thinking from that might have happened in the breakout rooms as well, just for us to together think about some of the key unanswered questions that remain in transformation and think about the opportunities that exist based on some of the research progress we heard about today in terms of new technologies and approaches, as well as, you know, thinking and discussing the challenges that might remain. Zeb, do you want to kick us off on this?
Sure. So there are a number of themes that I got pretty excited about throughout the day, but I think I'd like to focus on one that is near and dear to my heart, you know, which is thinking about how the spatial arrangement of cells is coupled to their behavior. And so cutting across all the presentations was this theme that the spatial arrangement is somehow important. And you know, the real focus there is that sets up the lines of communications between cells, allowing some cells to get signals, allow them to proliferate or otherwise. But sort of sitting underneath that question is, I think, is a really big open question, which is when you see the spatial arrangement of cells change, why is that? And so we saw a couple of lenses through which to think about that question. So it could be stochastic, in the sense that cells can make decisions and randomly end up in a certain place. And occasionally that new place will put the cell into a niche where it can overgrow. And we also saw examples, for example, thinking about Adam's work, where changes to cell adhesion could allow cells to end up in new places. And so I think a real opportunity for us is to think about ways that those two views can be linked together. Perhaps through the signaling molecules themselves or the oncogenes themselves that are somehow involved in these processes. And so is there, you know, is there some sort of a rule there or a set of rules that we can begin to think about to learn how these new spatial arrangements emerge, not in a completely stochastic way, although maybe it is that way, but perhaps in a more deterministic way downstream of these mutations that the cells are contributing or the cells that are moving into the tumor.
Yeah, I think the idea as well that there are tools and approaches that let us control that and try to recapitulate spatial organizations that we observe in clinical samples, right? And then to move back into the in vitro setting and give us a perturbable system, right? That's in my mind, one of the most important things about a preclinical model is that we can perturb and measure, which gives so much information. And just playing on that, one of the connections that I put together a little bit is how to bring some of the things from today, for instance, and you already mentioned Adam's talk, thinking about how to engineer the receptors to help modulate cell adhesion and perhaps look at phenotypes such as motility. And then we heard in the next session, Carolyn's talk, where she has these beautiful approaches to manipulate the internal molecular machinery. So, for instance, to introduce an oncogenic mutation into a single cell in a defined location gives us an opportunity, if one were to bring those two approaches together, to think about experimentally manipulating both the cell intrinsic and cell extrinsic relationships between cells. And then I imagine that Arthur Lander will model all of that for us to help us identify some of those, you know, which are the key changes that might modulate cell growth, you know, tumor growth, cell proliferation, and so on, using some of the computational frameworks. Eddie, you are nodding your head yes. So I feel like, I mean, and then a part of this as well, of course, is the fibroblasts. We didn't get a chance to hear that much from you today, but I think your thoughts on how that fits into this as well, I think would be great to hear.
So obviously, in a very biased manner, I was missing fibroblasts today, but the reason is not because I studied them, but because the interstitial environment of solid tumors is really made mostly up of the interstitial matrix, which is collagen-based, and fibroblasts are the main producers of this. The entire tumor mass and per niche, it is altered based on all of whom they are together within that. So cancer cells and other types of cells, even before cancer occurs, are responsible for altering this interstitial environment, yet the matrix itself is generated and made by the fibroblast themselves. And so to my little very self-centered world, that is what should be the basis of the environments that we're trying to mimic and model, because all of the cells reside within that, especially in the cancers that I am studying right now, which is cancers that go through a delamination very early. So even the physical interactions are directly between the epithelial and the mesenchyme component. And so adding that, and we spoke that in our breakout room, you know, what is the minimum complexity needed to really try to understand a question? Are we, and very importantly, recognizing what the limitations of our models, either computational or experimental, are, because what we cannot learn from what we're doing is probably as important from what we can. And we need to recognize both sides so we can move forward with each question. So again, the matrices and the fibroblasts, or thinking about the cells as units, as opposed to individuals. So we're all very fascinated with single cell studies, but a single cell is not a by itself entity. It is based in a functional unit that is biomechanical and biophysical. So each cellular unit with its own microenvironment or nanoenvironment, if we're talking about a single cell, is as important as the interactions that we were talking about today. So I don't think we're answering any of these questions. I'm just bringing more questions to the table. But I think that, as we mentioned before, no one of us will be able to do this alone. And that is why this is so important.
Yes. I did see that Arthur just raised his hand. I don't know if we have the capability for you to unmute and join the discussion.
I tried to unmute. Did it work?
All right, there we go. Yes.
Yeah, no, I just wanted to underscore something that Etty said about defining units, because really the only time in science that you successfully cross scales is when you can really define a unit at the new scale. And, you know, in systems biology, there was a point at which we defined this unit of the sort of network motif. You know, so if you wanted to jump all the way to big, huge networks of thousands of genes, it was just impossible, right? So at a certain point, someone said, okay, let's just jump to something we can understand. And then we're going to make the provisional assumption that everything can be composed out of units like that. And so then people started talking about, you know, type 2 incoherent feedback loops and things like that. I think we need the same thing in tissue biology and in cancer biology. We need to be able to say, at what unit can we say, this is really a unit of tissue function, and this is going to get repeated. And if we can just get that under control, then I think we have a pathway to moving forward.
Yeah, I agree. And there is a lot of biology is repeated and is patterns. And I think at this point, related to that, there is a lot of image data available, and now these incredible image-based analytics, right? The AI approaches that are exquisitely tuned for finding patterns in image data. And so I think there's ways of systematically defining some of those units as well, you know, from a data-driven perspective, as well as from a more prior knowledge, biologically informed approach.
One possible danger there, though, is to take abstractions from say, electrical engineering or computer science and try to force them upon these complex and messy biological systems. And so I think it's really important for us that we approach this challenge that Arthur has just raised for us from a place of naivete and really ask, what are the relevant dimensions of this problem? How do we really think about it in a way that is not simply trying to take things that we already know and apply them to the systems?
Yep.
I'd like to switch gears a little bit and talk a bit about dynamics, and really, I think for Ken and Bianca to get your thoughts on predicting dynamics. And in particular, Ken, at the end of your Q&A, there was some discussion about how do we even define a precancerous lesion? Well, it is precancerous because it eventually turns into cancer. And so there's the idea then that we need to be able to predict that sort of temporal trajectory, which we can model systematically in the lab, try to recapitulate that on the preclinical side. But I think there's also opportunities for thinking about how to mathematically model that. And would be interested, Bianca, to hear some of your thoughts on that general concept, sort of broadly, and for you to interpret that as broadly as you wish. And I see a note from Ken that he is muted, so hopefully Bianca is also able to unmute as well, or might need help. There we go. I see you both.
I can unmute, and until he's unmuted, but I can help answer this question. I mean, this is a question that I think a lot about. So we brought up effective functional units, and I think effective functional units, or modes that allow us to explain and rationalize about spatial organization, they don't have to be static. So to me, when I think about functional units, I think about dynamic functional units because there's a trajectory that we need to explain. And there's this really useful concept in statistics that has to do with spatial point processes or spatial marked point processes, where you have events with particular properties, and you want to ask the question, are they synchronized? Are they stochastic? Are they deterministic? And I think one of the things that came up to me in a couple of the talks, Caroline's, Josh's talks, we want to understand, okay, what events are truly stochastic? What are the events that are cascading from one another? Can we differentiate between these two modes of temporal progression? Because then we can maybe control them in an organoid or like in another vitro system. So to me, there are tools in statistics, but I think to make them truly applicable, as Zev said, we need to actually think from first principles that are relevant to the biological question at hand. It's never going to be possible to just take a tool and apply it and hope for the best. And I think we just need more conversations like this to bridge that gap. I don't know, what do you think, Ken?
Yeah, I think, a major, I think, dynamics, the temporal component, right? It's extremely important in systems biology, right? If you do an ODE, PDE, that time component is always important. And I think, an important, under-discussed factor with these in vitro model systems that we really want to get to is that actually we get to watch events in real time, right? You can think about using some advanced imaging techniques like 3D tomography and hollow tomography and things like that with AI, that you can watch cell types go in and out of a tumor microenvironment. This is actually very hard to do even in mouse models, right? Like we do some of that, building a window in a mouse, trying to do intravital imaging, but those are very limited, right? So having the ability to collaborate with bioengineers and synthetic biologists to actually get an in vitro model up and running that you can observe with the tumor microenvironment is actually extremely valuable, in my opinion. So with that said, I see a lot of ideas on Miro about this temporal component, about, hey, how can we model aging? How can we model clonal expansion? in vitro, etc. And there's actually one particular comment that I found interesting, saying that academia limits our ability to actually do this because it is a big problem and it's a big team sport, right? In order to build a realistic tumor microenvironment. And from today's talks, right, we can see that there are a lot of individual tools built to sort of engineer tumor microenvironment, but again, addressing specific biological problems. But then how that would recapitulate a real tumor microenvironment, and then the dynamics of it will require some large-scale bi-directional discussion between the data generator as well as, you know, the in vitro modelers.
Can I add a little bit to what Ken just said?
Yes.
So Ken, you just hit the nail on the head, because that's exactly what we were discussing in the breakout room 2. The fact that academia, not on purpose, is limiting us because we need a lot more complicated, and we're still working on a siloed type of system where each of us has a lab and each of us is driving something, and sometimes we collaborate with others, but it's still a system that is not putting all of these ideas together. And these days, with the technological advancements that we have, we should also be able to put them, to break these silos better. We need to change the system. So we were talking about that as well. And then Laura was mentioning that as long as we select each question one at a time, we might be able to do so, because we cannot answer the complex questions, all of them at a single time. So that was the type of discussion. But on top of all of what you just said and I commented, there is a feeling when we talk about progression and longitudinal changes and dynamics that we're still talking about plasticity, and we're forgetting biologically evident elasticity as well, meaning cells can go back and forth on their states and not just have a directional. So one of the things that Zev said is not to try to fit something into a box of something else. And so a lot of times, because we understand that in immune cells there is a full differentiation path, we tend to think about that for the dynamics of the cells. And the fibroblasts, for example, can be very elastic in their function, and their functions are not mutually exclusive, for example. And not until they are fully differentiated, meaning post-mitotic, that they will stop doing that. And so, and there is very little evidence of them actually doing that and persisting in the tissue. There is 3 or 4 each time, and yet the entire tissue is changing. So there has to be an equilibrium that is elastic and not just plastic. And so I just wanted to bring that up.
We're almost out of time, but Lucas, I want to give you an opportunity to maybe give us a short, 30 seconds or 1 minute thought on some compelling questions or ideas that you thought came out of today before we break.
Sure, thank you. No, I really enjoying the discussion, and I think something that came up in our breakout room, but also is echoed here, this kind of need for every big consortia to come together to sort of really just define what needs to be done. Because I think what we've seen in the talks today, from Carolyn's talk, and the technologies are extremely sophisticated. We didn't really, Mo mentioned a little bit about optogenetics and obviously genetic engineering. As an optogeneticist, anything you want to control dynamically with precision inside of a cell, any kind of biochemistry is possible. A lot of it's been done, and anything else, it's actually not that difficult to do. So a lot of these, synthetic approaches to rebuild communities and to toggle them this way or that way at this point or that point in time is doable. I think what's from the technical perspective, I think what there's this I would love to see is community-wide prioritization of these are the most interesting targets. It's the fibroblasts, right? It's the mechanics. Like, this is what we need to, the parameter space we need to explore more deeply. And obviously there's many of these there's many targets and many parameters, and it's kind of, and that's kind of the problem, is that it's a huge space for any one person to even think about entering. So I think that this kind of, I don't know, I'm excited to sort of to get that underway, because I think that there's clearly a lot of potential.
Yeah, excellent. I think that's a great way to end, because the idea really in these sessions was to bring different people together, ideally to break down some of those silos that we are all together in the same virtual room, and to start to think about what are the high priority questions, because for sure, if we want to try to go after new areas of research and address big compelling questions, then we need to have some focus as well. So just as a final wrap up as well, our plan is to take the ideas that came forward in the Miro boards and notes from today, and to incorporate those into a white paper summary of this workshop. And just the last reminder that this is day one. There's another workshop on Wednesday, and as well next week on therapeutic resistance. And we hope you all will join us for all 3 sessions. Thank you so much. And thank you to all the speakers and session chairs as well.
Thank you all.